topic. I would like to share my immense pleasure that I attended the first IPC in 1985 in India, inaugurated by the then Prime Minister Sri Rajiv Gandhi in the Siripur Auditorium. That was in 1985. That was the 37th annual IPC. And today here I am speaking at the 74th IPC. Now you can already start counting what my age is, like I'm a very old guy. That's 40 years ago, interestingly. And this is very humbling experience for me to be here to be the first speaker at this scientific symposium, the 74th IPC. I also want to thank a number of IPC organizing committee members, especially Professor Dr. Operant, who has been coordinating with me for almost, I think, 2 months. So, with no further delay, the topic that I am presenting today, our topic is on challenges and opportunities in the oral delivery of macromolecules in the AI area. So, my presentation is around what the need for macromolecule delivery and what the challenges are, how we do it today, what some of the improvements that have come.
Check.
The slides are not changing. So I cover my presentation under this the overview of the subsections under which I will be discussing my presentation today. Alright, what are macromolecules? Obviously, macro is large, so it's a complex structure composed of many small repeating monomers. And there are 4 main classes.
Now we are live.
Peptides, these are anywhere from 2 to 50 amino acids. Examples include insulin and GLP-1. Proteins, that is long chain greater than 50 amino acids, like enzymes and monoclonal antibodies. Nucleic acids, especially the mRNA, the COVID vaccine everybody knows was based on the mRNA vaccine. Carbohydrates like oligosaccharide and polysaccharides, I will have one case study or example that I will be discussing as well. Lipids is the other macromolecule that we are talking about. So, what's the problem here? The fact that there are almost 600 proteins or peptides in development globally. In development, this only peptides and proteins doesn't necessarily include the other classes of macromolecules that I talked about. And the market size is projected to be around $400 billion just in this space. And almost all of these molecules are essentially developed as injectables. That's a challenge. An oral version that works for these could be a blockbuster of blockbusters, especially for the anti-obesity drugs that are being developed. The source for the information is listed at the bottom there. Sorry, the slide motion is very slow. So, why pursue this? Oral delivery. I mean, patient impact, number one, right? Like the convenience. Have you heard trypanophobia, what that is? Yes, so fear of needles, right? I mean, that's what prevents the compliance and the convenience. So by developing these molecules in the oral dose form, we are able to address the main challenge. And also the self-administration. I mean, a lot of the parenterals of the injectable versions also require hospitalization. Not that everything would go in the like self-administration like insulin. That's not the case. I mean, a lot of them also require hospitalization, which is quite expensive. But now, the challenges. I'm sure you know the Lipinski's rule of 5, which talks about what are the ideal properties of a molecule for oral delivery. The log P should be less than 5. Molecular weight should be less than 500 Daltons, no more than 5 hydrogen donors, no more than 10 hydrogen acceptors. Those are some of the rules that I can recall. recall, but these molecules fall well outside of those properties. So that makes it challenging. So now let's look at what the main challenges are, how the gut biology that evolved over the 300,000 years ago is built to destroy any foreign protein. That is the main challenge that we are trying to address. Why the macromolecule delivery is a challenge, particularly the proteins and peptides, is the ability to bypass these hurdles. And what are those hurdles? Number one, acid bath. Body, as I said, human body is designed to destroy foreign proteins or peptides. Whatever therapeutic proteins we are administering, body is taking it as foreign material. Then acid bath, low pH in the stomach destroys a lot of these proteins. And then the enzyme reduction, which means like pepsin, for example, can destroy insulin in less than 30 seconds if it is not protected. And then the great wall or the epithelial barrier, it's like just a simple analogy, passing it through a keyhole. That's what we are trying to do because there is not enough gap. Paracellular and intracellular permeability is not possible with these large molecules. If you see on the screen, I don't know if you can see on the right side, pointer is not working, let me move fast. Then the permeability challenge is the biggest of all. Even if you solve those other three, the permeability challenge still remains to be the biggest hurdle for these molecules to be delivered orally. Size, polarity, these are mostly hydrophilic. And now you have to pass through the mucus layer and lipid barriers for hydrophilic molecule is going to be a challenge. Efflux is another problem. The body pushes those out even if they go inside. And the hydrophilication, which we talked about. Sorry, my computer is sitting there, so it's taking some time for the transfer of information. So, having said this, with all the challenges as well, oral macromolecule delivery is no longer a hypothetical concept. There are products out there that I would like to discuss today. Come on. So, there are ways to achieve this, and there are technologies that are out there that leverage some of these existing ones, especially the lipid-based formulations, and also in the Zydis technology, which is a lyophilized tablet that is sublingually or buccal delivery, which facilitates the delivery of macromolecules. So there are a number of approaches. This slide is very busy, but if you see around this circle, there's chemical modification of these peptides by attaching a lipid link to these hydrophilic molecules. Addition of effective agents that includes penetration enhancers or formation enhancers, mucolytic agents, proteolytic enzyme inhibitors like APTI or the soybean trypsin inhibitor, absorption enhancers we talked about. Then the delivery mechanism. You see on the left side there, the up-to-gel DR or the delayed release soft gels is an approach where you can deliver the molecule to the site of absorption or where it is much safer for the molecule, like not in the acidic zone, but drop it in the more neutral or basic pH area. So those are the four main pillars. Then the medical devices. I don't know if any of you heard RANIPIL, RANIPIL. So that's supposed to inject into the membrane directly. You take it as a capsule, but it then injects the medication in the body and then body gets rid of it, which is a novel technology. So these are some things that are happening, and of course this is still not officially available yet, but it's under development. So there are more molecules that are viable for delivery than people think, or the formulators or the scientists think. I mean, these are the categories, peptides, especially the 1 to 3 kilodalton. Of course, most of the newer versions, the GLP-1s, they are higher than 3,000, so that is where the challenge comes, of course. Peptides, we take cyclosporine is a peptide with 200 dalton, voclosporin is a peptide. They are all delivered today orally. and polysaccharides like sulfide that you see there is 7,000 Dalton molecular weight, and it is available as an oral soft gel in lipid system. Then the formulation of molecules that benefit from enzymatic protection could also be delivered by this approach, even if they are macromolecules. So, I think I missed a slide there. So, if you had to justify cold chain, I just give one example quickly. I worked on a biologic molecule. Cold chain from API manufacturing all the way to delivery in vial to the hospital for administration will cost $10 US I'm talking about. But if you convert that into room temperature, which we did successfully, we could cut the cost to 60 cents from $10. That's the kind of other benefit that you get by converting these injectables, especially requiring refrigerated condition to room temperature. And that's possible, and we have a number of examples that we can discuss today. So, how are we breaking this injection barrier? We are breaking it through at least some of the technologies that we at Kaplan work. One is the lipid-based soft gels as armored escort. What we do, again, I'm not going to read in the interest of time. There's a lot here, but you can see on the left side, it is enteric soft gels, protect from pH, enzyme inhibitors, formation enhancers, microemulsions, like if you take neoral cyclosporine or voclosporin, how they are delivered. And that enhance bioavailability for anywhere from 2 to 40% with these approaches that we've already, and these are proven and already in the marketplace. And on the right side, Zydis ODT, this is a wafer thin, 1 to 3 second dissolve. These are prepared by lyophilization technique. These are tablets, but lyophilized tablets. So 1 to 3 seconds sublingual or buccal delivery, and which facilitates and avoids some of the challenges that these molecules face in the gut, because there is a gastric absorption that takes place when you are able to deliver it sublingually. So, lipid-based formulation, how formation enhancers, surfactants, the cetyl fatty acids that we use as vehicles in the soft gels actually get converted in the body into a formation enhancer, forming sodium caprozoate, and that acts as a formation enhancer. And then the surfactants break through some of the mucus layer that is preventing these molecules from getting into the body. Sodium caprate, we talked about this, and the way it is available to make a tablet. It's a very crystalline material, very difficult to make tablets, but in the form of medium chain triglyceride, it in situ, meaning in the body, it gets converted into the C10 formation enhancer. That's what we leverage. So when you deliver medium chain triglyceride, it gets converted into mono and diglycerides, and those act as formation enhancers. So as I said, there are a number of molecules that are delivered clinically already, meaning they are available, which are macromolecules. Desmopressin, and again, it's not just soft gels. If you see on this slide, there are 4 different technologies. One is Zydis on the very top left side, which is a lyophilized tablet that delivers desmopressin. Molecular weight is over 1,000. Semaglutide on the right side is delivered today using a snack that is a formation enhancer by Novo Nordisk. It's in the tablet form. Bioavailability is only 1%, but it's still doing the job it is supposed to, and it already this year did $4 billion in sales. Cyclosporine, 30 to 38% bioavailability. It's a self-microemulsifying drug delivery system. And on the other side, voclosporin, it's the same group or same family. The reason these work well is these are cyclic peptides, whereas the other ones are linear peptides that are much more prone to destabilization by the gut system. And sulfide there that you see, I show it on this slide better. 7,000 molecular weight, close to, and oral bioavailability is 40 to 60%. granted that it's not a peptide, but the fact that it is a very large molecule still getting delivered and making bioavailability almost 40, 50 or even 60% is very interesting. So lipid-based systems essentially exploit the body's natural mechanism or ability of our body to digest the lipids. When the lipid is digested, the drug it is carrying gets dissolved or made it more permeable, and there are two approaches: either use long chain fatty acids or the short chain fatty acids. When you use long chain fatty acids, they avoid the liver plus pause effect and get delivered directly into the lymphatic system, avoiding the damage caused by the liver plus pause and enhancing the bioavailability. And the short chain or the medium chain molecules facilitate the systemic absorption. So here are some examples. This slide is again very busy, but the versatility of lipid-based formulations is explained on this slide. Depending on the physicochemical nature of your molecule, you could actually select one of these approaches within this family of lipid-based systems, whether you use a simple triglyceride or a combination of hydrophobic surfactant or a combination of co-solvents, hydrophilic solvents, and surfactants combination. So this wide variety or the versatility of lipid-based formulations is what made possible over 70 prescription drug products approved by US FDA, and almost 67 of these were developed by various pharma companies, big pharma with Catalent, and these were approved out of Catalent sites over the past 50 years. And if you look at the other thing I want to point out on this slide is all classes of drugs are delivered using this system. PCS 1, 2, 3, and 4. 4 is the one that is fully soluble, fully permeable, and then you can connect the fact that your peptides or macromolecules are fully soluble. That's where we leverage this, and if you can see here, octreotide, it's a PCS4, it's a peptide, and it is delivered in the form of a lipid-based formulation commercially available. So this one, again, the point of this slide, the take home from this slide is, it's not as simple as just mixing oil and some surfactant. You need to understand the dynamics of how the ratios of the various lipids and solvents and surfactants that use can make or break your formulation. In this example, only formulation 1 produces a thermodynamically stable microemulsion, not the other one. So it's very important to understand the science behind these formulations. So this slide, again, very busy slide, but two things I want to point out simply. On the left side is type 1 and 2, right side is type 3 and 4 type of formulations. Obviously, they are forming microemulsions because it is a combination of, if you remember from the previous slide, co-solvents, hydrophilic solvents, and surfactants, whereas the left side one is mainly lipophilic solvent or lipophilic surfactant. That's what makes it different. And also, depending on the type of fatty acid used, long chain fatty acid, something like C16, C18 type fatty acid, it results in liquid absorption through the lymphatic system. So, what are the other innovations that we have that are being utilized to deliver the macromolecules? On this slide, you see two different types of soft-gelatin capsule. The first one that you see there is totally made of vegetarian or vegetable plant source material, which is carrageenan based, and the one at the bottom is made up of gelatin combination either by coating, entering the very first one that you see there, or this one is the newer version, which is a combination of gelatin and pectin, that in a single step you can manufacture this, reducing the cost involved and the wastage. And on the right side you can see various release profiles we were able to achieve. Again, those release profiles are controlled through the film matrix system, and the shell can delay the release. For example, you see this one here for 2 hours slow release or 1 hour in low release, whatever you need, you can tweak that composition of the shell to achieve that. And here are some results from this in the monkeys, non-human primates. 8 hours is when we achieved the Cmax, which means these could be utilized as functional shell, not just regular shell. So here is a good example of how the formulation of a calcitonin, which is a $3,400 molecular weight peptide. It's available as a subcutaneous injection. Delivering it through subcutaneous injection only gave 21 to 53% bioavailability, whereas even with the soft gel capsule as EC, that is enteric soft gel, gave almost 39 plus minus 12. So nearly achieves the subcutaneous level of bioavailability using right formulation again. Key is, and the same one delivered only in an immediately soft gel, give only 10% bioavailability. So that's something to be noted. So quickly, time-wise, how am I doing? I didn't check when I started, so just alert me. Another example, higher peptide bioavailability than tablets. This is a peptide that's around 5,000 molecular weight, a powder pill capsule with the formation enhancer versus a delayed release soft gel liquid system, resulted in almost 24 times higher bioavailability using this approach in a dog study that was just completed last week. So it's not that all peptides will benefit from the GI delivery of the lipids. So there are some molecules that will benefit from pre-gastric delivery using the sublingual or buccal. So you have to see your molecule nature, but at least there are a couple of technologies that I'm talking about today. One we talked about is the lipid-based system, and now I'm talking about Zydis, which is a lyophilized tablet. And why it helps, as you can see, the buccal or sublingual delivery avoids mainly the gut problem that I explained in the very beginning. High pH, the low pH utilization and enzymatic degradation. You are avoiding all those by facilitating some pre-absorption or pre-gastric absorption. And there are 4 marketed allergen products in Europe and US. These are for pollen allergies, for example. These are 50,000 molecular weight, $50,000, huge. And they are delivered using this technology successfully, and more products are coming. And here is another example of influenza vaccine delivered using this system. And I'm showing this slide is very busy, lot to read. So let's make it simple. We tested 4 or 5 formulations. F8 that you see here was the best performing. And this is the clinical disease score, like meaning almost no disease treated with this. And in the side panel, you can also see untreated animal, almost 20% of those had the disease or died, survival rate, whereas the one treated with this system survived almost nearly 100%, 95 to 100% protection was offered by this system. So now we talked about the old paradigm, which is essentially you are taking a molecule, trying to escort it, trying to give protection to enteric through enzyme inhibitor addition, formation enhancer addition. All of these are take the existing molecule and somehow get it delivered, right? That's the existing paradigm. The new paradigm, the AI, you're actually modifying the system or the molecule itself so that you identify what are the functional groups of the amino acids that are causing the problem, that is destroying the molecule. How about if you replace that? So that's where it is going. So before I go into that, AI is impacting everything we are doing already, right? Starting within R&D, we today use AI agents to write protocols and development reports. It takes some effort, but if you do that, and it cuts your time by more than 50%. And a company like us, we are generating over 5,000 to 10,000 protocols and reports each year globally. I have a team of 1,000 scientists across 20 sites in 4 continents. And by using some of these techniques of the AI, we are able to cut the speed, efficiency, and the predictive power, cost savings, and regulators are adopting it. If you look at regularly, there are updates coming from FDA as well. How they are adopting AI in the development as well as in the review process. So again, there are a lot of numbers there. In the interest of time, I'm not going to read all these numbers. You can see it for yourself. So when it comes to developing oral peptides, as I said, barrier is acid bath and enzyme studies. What is the AI strategy? Molecular redesign. You are designing the molecule to avoid these two problems by eliminating the functional groups from the molecule that are causing the problem. How is it being done? Using generative AI, protein language models, and diffusion models. That's how it's being done. It's actually happening, by the way. There are several companies in the US and other countries that are doing the Great Wall, epithelial barrier. Again, smart formulation. You check various permutation and combination of. In one of the slides I showed, I said 24-fold increase in bioavailability, but there were also 6 other formulations that were not doing anything. So the key is identifying what works and what doesn't work, right? That's what you are trying to understand. And by using AI, you are able to leverage that much quicker. High variability, patient to patient. So digital twins, what is that? Each of us have unique, even though our system is same, but some may have a different pH in the stomach in a different zone. So this maps out everything, and performing an APTI modeling, you create a digital twin. Before even you administer your molecule, you know what works and what doesn't work. That is how you are actually managing this. So, I'm almost on my last 2 slides. Hopefully, time-wise we are still okay. As I said, AI is being used to design or redesign the molecule that retains the potency but resists proteolytic enzymes and pH. Recent breakthrough, Microsoft actually, this you can find on the Microsoft website, tangent model, where they used the generative AI, developed target aware molecule generation. Means the molecule knows what is my target and where I should go and work and how to avoid the pH problem, the enzymatic problem, and formation problem. And then the diffusion models, again, you can, it's a big, there's a reference I put out there. It's look into that to understand what that is. Not enough time to explain that, but that technique is leveraged to address the proteolytic problem and the formation problem. And 2024 Nobel Prize in Chemistry was awarded to those gentlemen there who validated that it's possible now to computationally generate these structures of amino acids or peptides, proteins. So those are the current developments in the AI era that we are talking about, how to develop macromolecules. That is already happening as we speak. So again, AI adds value in all these areas, formulation optimization, doing it in a fast and quickest way possible. For example, 10 to the power 5 formulations, it could predict their performance 10 to the power 5 in a matter of 1 week. And if you have to do that, it would take you a couple of years to do that many formulations. That's the power of the AI that is being leveraged. Predictive modeling, again with the data, AI is all about data, and you have big data sets to come with a model that works, and manufacturing robustness. And the key takeaways. So, oral peptide delivery is not a dream, it's a reality, it is happening, and it's possible. At least I presented 2 technologies, it doesn't mean these are the only 2 technologies, there are, I'm sure, other technologies out there, but these have made possible all the examples I presented today, except for the semaglutide example. All of the other examples are using these 2 technologies today. Oral delivery can expand indication and performance in compliance, as you heard, and lipid-based formulation in soft gel and dry solidity expand the development of macromolecules in a significant way. We continue to do that, and we are working on a number of molecules in this space with these technologies. And I don't know if there are any pharmaceutical companies in the room. Can someone raise your hand if you are from a pharmaceutical company, or all academics only, or anyone from pharma companies here? No? Okay. So what I'm trying to implore is don't not consider. Make sure you consider your options before you decide. Don't go with what is easy and what is available in your company. Like, okay, I can do tablet easy or hard capsule easy and only go with that. No, that's not the way to do these molecules. For these molecules, you identify the most appropriate and proven technology. That's what we are asking. And with that, I will conclude my presentation, and I want to thank the IPC organizing committee, as well as all of you who are sitting here patiently. And thank you again. Any questions? You're absolutely correct, sir, and that is actually a new drug. If you see the word that I used, molecular redesign. So now you are designing molecules that are intelligent to know what their ultimate receptor is, where they need to go, without getting destroyed along the way. Right? So they are indeed new molecular entities. So for the existing ones, we are trying to convert them. As you see, cyclosporin is a good example. It used to be an injectable in the 1970s. The first they converted into a liquid solution in a bottle, then a liquid sanitary soft gel, and later a neoral soft gel. So, but you're right. These are new molecular entities that are coming using this. Correct. Yeah. Yeah. Any other questions? Someone raised a hand this side. Can you give the mic to him?
The gentleman is talking about functional groups.
Yeah.
Removal and acceptability or not acceptability.
Right.
But is it that all functional groups are responsible for therapeutic activity?
So, not necessarily. I mean, you know, at the end of the day, with this AI redesign of the molecule, we are trying to ensure that it is still therapeutically effective without getting destroyed along the way. Right? So, not every functional group is doing the therapeutic job, but there are certain functional groups maybe facilitate their permeability, some solubility, and some chemical stability protection. So, the idea with the AI is to look at the whole spectrum of molecules that are currently existing, that are researched, hundreds of thousands of them, right? From the discovery phase they are looking at, and not every company can go and do this. I mean, obviously there are companies.
It basically connects all data points.
Yes, exactly. Yeah.
And one more question, sir.
Yeah.
You talked about entry coating.
Yeah.
Is it by applying polymer coating or any cross-linking?
So there are two capsules I showed there. One is entry coated. This is duplication or manufacturing step, like you manufacture the capsules, then take them and pour them similar to tablets. The other approach that we have designed is combine gelatin and a biopolymer, and it forms a reversible cross-linking that is pH dependent, meaning in the acidic pH it is intact, it doesn't open. When it's in the basic pH, those cross-links are broken and the drug gets delivered.
This is very complicated, critical cross-linking.
Yeah.
If it exceeds, it won't dissolve.
Correct, and that's where the challenge, and that's where we come into play with this new technology.
Good.
Well, if there are no questions, then I think Dr. Karunakar has done a very wonderful job, and so nicely. I think it's they are the two sides of the pie. The first side, you made us understand as to how macromolecules are very important, and there are lots and lots of newer macromolecules, more than 600. And then you also told us, the students and the younger generation, that lipid-based systems are easier to go ahead.
Yep.
And then after that, in various publications and hard data, comparative scenario, can you look it with the artificial intelligence? How you can quicken the process, how you can hasten the process of optimization and arriving at the right kind of molecule, whether it is an in vitro study or perhaps an in vivo study. I think our younger generation should take time. The computational pharmaceutics is advancing very, very rapidly. You can do in one day what we could do in 1 year. That is the state of improvement which is expected in next 5 to 10 years. This lays down a very good sort of message to all scientists that we in India should be able to do it and start the process with step one. Thank you, sir. Congratulations.
Thank you so much.
So lovely, and I've learned so many things, and I'm sure your education teachers will take away, and you have also said many takeaways. They are also good. Thank you very much, sir.
Thank you so much.
Thank you, sir, for the enlightening session on oral delivery of macromolecules in the AI era. I would request the chairs to hand over and felicitate the speaker, Dr. Sukru Karnakar, with memento. Thank you. We are moving on to the second scientific section by Dr. Tuan Lamas. Dr. Tuan is Chair of Experimental and Molecular Imaging, RWTH, Ancient University, Germany, and he has expertise in imaging techniques, especially in tumor targeting and smart cancer nanomedicine. I would like to request Dr. Tuan to come forward to the stage.
Can you hear me now? Yes, that sounds good. All right. Ladies and gentlemen, dear colleagues, I'm going to discuss with you today how we use advanced technologies based both on artificial intelligence and machine learning as well as imaging to improve the performance of cancer nanomedicine. So advanced formulations that I guess we all agree hold intrinsic potential to enhance both the efficacy and tolerability of anti-cancer medications, but where we're struggling to actually make an impact on real day-to-day clinical practice. So, if you look at the cancer nanomedicine formulations that are at this point in time on the market, approved, used in the clinic in patients, if you're very strict on definitions, these are the formulations that we are currently having. So in this overview, I excluded things like antibody drug conjugates, etc. Many of these formulations are based on lipids, some of them are based on proteins, two of them are based on the organic compounds, and one of them, in the middle panel, Vixios, is what I think one of the next generation nanomedicine that really hold potential because they combine two drugs in one nanoparticle at an optimal ratio. Right? And if you would give those two drugs separately, they would of course distribute completely differently in the body, only by using nanotechnology to co-deliver those drugs to a tumor compartment, to the tumor cell, you can exploit that pharmacological synergy. What we've been observing, and this is already a fairly old slide, but people have been becoming increasingly critical of the cancer nanomedicine field. I think there are a lot of papers out there with a lot of strong claims on how nanotechnology completely changes the way we treat cancer. But I don't think we've been very efficient in translating these drugs to the market. Other people have asked the question, what went wrong with anti-cancer nanomedicine design and how we can make that right? And among the things that we've been doing in the lab to address it are these four elements. And particularly important for me, and that will be the first half of my talk, is the aspect of patient stratification. So if you look how new anti-cancer agents are being developed nowadays, right? Kinase inhibitors are only given to patients with specific mutations, for instance in EGFR. Antibodies are only administered to patients that overexpress a certain receptor. And if we make a new liposome or a new nanoparticle, we'll give it to everybody, right? Because we don't know how to biomark patients for stratification. And that is something that we've been doing for a whole bunch of years now, using different technologies. What is very important to note to start off with is that nanoparticle tumor targeting in patients does work. There are a lot of, maybe not a lot, there are a number of papers out there in the literature that state that this EPR effect, enhanced permeability and retention effect, that causes nanoparticles to accumulate in tumors, that only works in mice, that it doesn't work in humans, that's really wrong. Right? Like, there are a number of studies out there, and on the right you see one of the most comprehensive ones already done many years ago. I basically replotted all the patients. There are 17 patients that are administered with radioactivity labeled liposomes. You'll see some examples of tumor targeting here on the left. And all the way on the right you see that when you use SPECT imaging technology, 50% of patients are actually positive for nanoparticle activity. Right? So it does work. What you also see is, if you look at the numbers all the way on the right, is if we include patients that have high levels of accumulation, it's much more likely that they will respond. Right? So we could use imaging technology to do actually this type of stratification. I'll give you an example of a formulation that we developed in the Netherlands together with Christian and Wim Henning, which is based on core cross-linked polymeric micelle technology. So this relies on an HPMA lactate polyethylene glycol blockable polymer, which self-assembles into a micelle, and then these micelles can be cross-linked, and on the cross-linker, we have chemotherapy agents that release the drugs with predefined release rates. So the system is really optimized to cater various aspects. It may seem complex, but it's actually translatable. It's upscalable. This has been in many patients and it performs really well. These are some of the first results we achieved at the preclinical setting. So we compared these micelles to different types of nanoparticles. What you see in the images in vivo in the middle and on the ex vivo images on the right is that tumor targeting with these micelles is highly efficient. Right? If we do this with liposomes, granulated liposomes that are used, for instance, for doxorubicin delivery, like Doxil or Calyx, we typically achieve tumor accumulation in the order of 10% of the injected dose. With these micelles, we can go up to 20% of the injected dose per gram of tumor tissue. And what is key for this concept of doing imaging to actually select patients is that you demonstrate that the amount of tumor accumulation correlates with how efficient the anti-tumor efficacy is. And that's what we did in this study, in which we developed so-called theranostic micelles, which are loaded with a chemotherapy drug, paclitaxel, as well as with a tracer, which in this case was an optical dye, size 7. And what we then demonstrated is that over 3 weeks, over the course of 3 weeks in which we administered the micelles multiple times, that the higher the tumor accumulation is, and that you see on the right and the bottom, the higher these clouds, these concentrations in tumors are, the smaller the tumor eventually is, right? Because the tumor responds to the efficient delivery into the chemotherapy. From a clinical point of view, what is more important even is that by finding those tumors that do not accumulate micelles efficiently, we can predict which patients eventually will not respond very well. And then those patients can be excluded from clinical trials, basically how we do it based on genetic screening or receptor staging. We particularly also look at metastasis because many patients with metastasis are typically enrolled in clinical trials. That's the key reason. The key reason is that eventually chemotherapy is mostly used in patients with metastatic disease. And we really do not know a lot about what drives accumulation of liposomes and mast cells and antibodies in metastasis. So we developed some imageable models in which we can look at the one side on where the cancer cells are. If I go back, like in the middle panel, you see where the cancer cells are in the body of a mouse, with the primary tumor in the lower part, and then metastatic lymph node a little bit more up. And then we label the mice with a dye that can be imaged at a different wavelength. And then we can overlay those two, and we can actually figure out in which metastasis, and for which reasons these nanocarriers accumulate. And what we've observed is that depending on the vascularization and the collagen status of the mice cells, particularly the collagen status of the mice cells is actually determining. We noticed that in lymph node metastases in this particular mouse model, there's a lot of collagen cross-linking. That means the tumors become stiffer, those metastatic lesions, and it's more difficult for nanoparticles to eventually extravasate and penetrate into those cancerous lesions. When we then go back to, if this plays, skip this, go back to the human situation and to the in vivo imaging with radioactivity. So what we here developed is a theranostic for crossing polymeric mice cell, in which zirconium-89, this is a long-lived radioisotope, right? So we can then image for up to a week after administration, and docetaxel as a chemotherapy drug, or essentially cold-loaded into the same mice cell nano platform. And then we can monitor in patients, and you see two examples here on the right, whether the nanoparticles accumulate in those lesions. So in the top example, this is a jaw lesion of a patient, so you see the nodes on the top of the patient. You see that there is fairly good tumor accumulation actually in certain parts of the lesion. So you see that in the left part and in the right part, there is fairly good accumulation in the range of 5 to 6% of the injected dose. But it's not homogeneous, right? You see clear parts of the tumor that do not accumulate. So if there were enough patients to choose from, this would probably be a patient that you would want to exclude in a clinical trial. The bottom example is a must-include case. This patient had 2 lung metastases coming from colorectal cancer. You see the lesions outlined here in the middle panel with computed tomography, and on the right, you see the overlay of the mice cells radioactively labeled on those 2 metastases with accumulation in the range of 10 to 12%, and with a very homogeneous pattern of accumulation. So this patient is much more likely than the upper one to respond to this type of targeted therapy. And if you now look at all of these, and please just look at patient number 2, patient number 3, we can use imaging information to address heterogeneity in tumors. Patient number 2 had in total 12 metastases. And if you look at zirconium accumulation, it's 0 out of 12 lesions that accumulate. This would be a patient that does not make a lot of sense to include in a clinical trial because he or she does not accumulate the nanoparticles in the tumor, and he or she is very unlikely to respond to therapy. If you look at patient number 3, there are 2 metastases which both accumulate the mice cells efficiently. This would be a patient that you definitely want to include in such a clinical trial. So the good thing about imaging and advanced imaging technologies is that it's highly quantitative, it's highly specific, it's high resolution, it's very accurate, so it's very good in terms of specificity, but it's not good in terms of simplicity. Right? You need dedicated radiochemistry facilities in the hospital. You need to order radioactivity for every patient. You need to have companion diagnostic that is as safe as the inherent parental compound. So what we've been actually looking at is, can we find a compromise in which we take biopsies from patients which are available anyway, right? Because you need to put a needle into the vast majority of tumors. 99% of tumors are biopsied, because you need to first make sure that it's actually a cancer, right? And then you look at things like proliferation or receptor status, etc. And we were asking the question, can we look at biomarkers in those biopsies to identify which lesions are targetable with liposomes or not, right? And then you don't need any imaging, right? And the biopsy is anyways already in pathology. So it's a very simple way to try to predict which patients are going to respond. And this is basically the study design. We looked at 23 different features in those tumors, related to the vasculature, to the cellular density, to different types of collagen, to macrophages, other types of immune cells. We did this initially in 3 tumor models, and then we validated the models in 13 additional models. We used gradient tree-based machine learning to identify in an objective manner which features are the most important, and we validated things with different types of nanocarriers. So what we first did is we generated data to train this machine learning model in 3 tumor models. For that purpose, we took a polymeric nanocarrier. We worked a lot with an HPMA polymer that is the size of an albumin, and that circulates really, really long. That achieves tumor concentrations in the range of up to 30% per gram of mouse tumor tissue. And then we actually went into 3 different models. One that has low accumulation, the one in the middle has medium accumulation, the one on the right has high accumulation. We did multiple animals per model, and then we evaluated in those 3 different models what biomarker features are expressed, and do they actually correlate with the accumulation of the nanocarriers. So if you just take the example for macrophages, for instance, you see that in the low, medium, high, you see a gradual increase. You see it in this bottom panel of macrophages. So low macrophages means low accumulation, high macrophages means high accumulation. Right, so this is already a good biomarker. You see a very nice correlation also in this bottom panel there on the right. And then we use this gradient-free boosting method where you basically leave one out and you retrain the model, you add that one, you leave another out, you train the model again. What we eventually ended up getting is 6 out of 23 features that are statistically significantly correlated with accumulation. Right? And out of those 6, you see those on the right, 5 were related to blood vessels, and the 6th one was macrophages. Which makes a lot of sense if you think about it, because any type of drug, right? It doesn't have to be nanoparticles, it can be an antibody, it's CAR T cells, small molecules, they need vasculature, functional vasculature to get to the tumor. And then in the case of nanoformulations, they actually rely on macrophages to take them up and to eventually also activate them. Right? Doxyl liposomes is activated in tumors by tumor-associated macrophages. So we identified vessels and macrophages as the key driving features. And then this is the key experiment. So here we took 10 different tumor models. On the top left, the highest accumulation. On the top right, the lowest accumulation. Each of those tumors we sectioned, and then we stayed 3 sections per tumor. And we gave the same sections to 10 different observers, which were blinded for the tumors, right? They didn't know what they were looking at. They knew whether they were looking at blood vessels or macrophages, that's the only information they got, but they were not informed on tumor background. And then they scored from 1 to 4, 1 being absolute low, 3 being 4 is high, and we made a biomarker score that is plotted. So if you have a low times low, that would be a score 4. If you have a high times high, that would be a score 16. And then we plotted the area under the curve, like the amount of doxorubicin liposomes that accumulate versus the intensity of the scores of those 10 different observers. And what you very nicely see is this differentiation, where the tumor models that have high doxyl uptake have a high score, so they correlate to high vascularization, high macrophage content. And what is clinically most important, we were very well able to identify the true negatives, which are those patients that, because of their low vascularization and low macrophage content, are not likely to respond to nanotherapy. And this is something that we're now taking forward in some sort of models to explore whether this type of simple but still quite specific patient stratification can help to better select patients. In the second part of the talk, I'd like to talk about priming. What can we do with advanced imaging combination again with machine learning models to address the fact that in a tumor, vascularization is often not as good as you would hope. And collagen content is often very high, which makes penetration. So, first example I'd like to show with you is we use an AI-enabled imaging technology. This is functional ultrasound imaging, in which we do we call this in-house bubble tracking. So in ultrasound, we use microbubbles as compensations, and we can predict, kind of like a GPS setup in your car or in your phones, we can predict where bubbles are at later points in time. So we basically see the functional tracks of blood vessel perfusion in tumors, not invasively, right? So we can, with ultrasound, we do not need to put in a needle or nothing. And then we get the velocities and we get the directions, we get the distribution of the vessels and the densities, and this really allows us to capture tumors that have different flow or velocity kinetics. And we do this, and this is a very important point, because what we see in the clinic, not so much in mouse models, but particularly in patients, is that many tumors have very bad blood flow. So this is an example from a patient that we did that had triple-negative breast cancer. It's an aggressive type of cancer for which there's no targeted therapy. And in the biopsy, we saw a lot of blood vessels. But then when the patient goes to the radiology department and you put an ultrasound transducer on the breast of the patient, on the tumor, what you see in this whole tall part, so this is approximately a 4 centimeter tumor, the whole black mass, only the part that is color-coded in the middle is actually perfused. Right? But that's 85%, 86%. So a lot of that tumor is not carrying blood flow. And then you have a lot of blood vessels that are compressed because the tumor has a lot of solid forces on the blood vessel. That means the tumor cells, they keep on proliferating and they gradually compress the blood vessel. That means the blood vessel is occluded. Right? And if you then take a bubble and you gradually oscillate it, you see that movement over there. So ultrasound is a pressure wave, right? So it will make an air-filled vesicle coated with a lipid or with a polymer. It will make it oscillate, and that will then reopen blood flow in this direction. Right? I'll show you an example in the cancer case first, and then later on I'll show you an example how such a moving bubble in a blood vessel can also open up the blood vessel in a transvascular direction. And that is relevant again, for instance, in the case of brain tumor treatment or in general drug delivery to the brain. This is an example of a real patient. It's a cat, so this is not an induced tumor model. It's a companion animal that had a tumor. This was done in the veterinary clinic in Utrecht in the Netherlands, where we monitored blood flow in a tumor. Right, so on the left, I hope the video plays, yes, you now see first perfusion before we do anything to the tumor. Bottom compartment very well, reduced top compartment not at all perfused. Then we apply 4 cycles of ultrasound with bubbles to open up the vasculature, and now you see how in the top compartment blood actually starts running. Right, and that gives us a window. Some people think this lasts for hours. My gut feeling is 5 to 10 minutes, and then it will relapse. But in those 5 to 10 minutes, you have a window of opportunity to deliver something to the tumor that can then cause a lot of direct tumor debulking. And if patients are naive to chemotherapy, if they've never seen any chemotherapy, they typically respond very strongly to the first doses of chemotherapy. And if you kill a lot of cancer cells, you basically reduce the mechanical force. Right? And then at the next dose, the blood vessel will not be compressed anymore. This is another example, also triple negative breast cancer patient, a different example than I showed you before. This is a 5 centimeter lesion, the dark part of the tumor. And when we look with Doppler ultrasound, you can see one blood vessel that is above the threshold, but it's outside of the tumor. Right? If it goes through the tumor, we don't know. We can figure that out if you play with the transducer. But the point here was, so we know we have very poor flow. And then every alternating minute, minute 2, minute 4, minute 6, and so on, we apply bubbles together with ultrasound to open up the vasculature from the inside. And then you see that after 10 cycles at minute 20, we now have more than 50% of the vessels in that tumor perfused. Right? Again, giving us that window of opportunity to get drugs, to get drug delivery systems into the tumor. And this is one of the very first studies that is actually done in the clinic. These studies are really difficult because they're typically investigator-initiated. There's not a lot of funding for this. You need a lot of technology and decent standardization to do this. But this shows that in principle this works. And this is, I would say, one of the worst cancers. If there are top 3 worst cancers, pancreatic cancer and glioblastoma are definitely in. Right? This is the pancreatic cancer example where you see in red if you do standard chemotherapy and in blue if you do standard chemotherapy combined with clinical regimens of ultrasound and bubbles that you normally use for imaging. So you're using imaging technology to enhance the chemotherapy response. Now, taking this to the blood-brain barrier, all of you with a pharmaceutical background know that the blood-brain barrier is really a formidable barrier to get drugs into the brain, right? Not only to brain tumors, but for many different diseases. And in the brain, we can do almost the same thing, right? The technology is even more complicated, so it needs a lot of also machine learning-based setups based on MRI and temperature mapping, based on these helmet-based devices. So this is basically a helmet here that can go into a magnetic resonance imaging scanner, and then you can focus the ultrasound energy in very tiny spots in the brain, and then using microphones, you can actually open very tiny spots in the brain, the blood-brain barrier. So we and others have been shown that this actually works quite nicely. You see an example there in the bottom. You see in the top, you see a blood vessel running through the brain. 70 kilodalton X-ray, just a sugar molecule that we use as a recorder, right? It does not leave the vasculature in the brain unless we do 3 cycles in 5 minutes of ultrasound plus microdose. And now you see how the approximately 10 nanometer X-ray is able to cross the vasculature and get into the brain. And what we try to figure out, so ultrasound is in principle extremely powerful, right? If you have a kidney stone, you actually use ultrasound to destroy kidney stones. So we need to do this in the brain very gently. So one of the things we did is we figured out at perfectly safe conditions, we optimized the dose and the type of microbubbles, we optimized the frequencies and the amplitudes of the ultrasound. In both examples on the right, for small antibody-sized polymers and for normal 100 nanometer size liposomes, you can see in green that the particles are actually able to get out of the vasculature into the brain without causing any side effects. So we looked at those mice, they behave perfectly normally. If we do pathology on them afterwards, there are no bleedings, no infarctions, no inflammation, etc. So this can be done under perfectly safe conditions. And then one of the things we're trying to do with this is to use this advanced technology. I guess we all realize that it will be very costly to do this in a clinic in a day-to-day setting. So it needs to be a very high medical need situation in which this adds value. And then one of the things here that we've been looking at with people from the Netherlands is what is called a diffuse intrinsic pontine glioma. It's a tumor that grows in the brainstem of kids, and it has a survival of 0%. So you see here for various different types of pediatric cancers, you see the long-term survival increasing. The IPCA stays at 0%. I think there are a number of reasons why that is the case. So it's located in the pons in the brainstem, which controls respiratory motion. So if you would operatively remove the tumor, you would basically stop breathing. Right? It's diffuse, that means it completely mingles with healthy brain tissue. That's why you cannot surgically remove it. That's why you cannot properly irradiate it, because it will kill too much of the healthy brain. And then the thing is, drugs like simple doxorubicin or HDAC inhibitors, they're super sensitive in killing those cells, but they don't get there. If you take them in a petri dish, it's picomolar concentrations of HDAC inhibitors kill those cells, but they don't get there. So we need to do something, right? We need to use this nano-chemotherapy combination to actually open up the blood-brain barrier, and then we use the nanoparticles here to do what I said in the beginning, to combine two different drugs in an optimal ratio to hit cancer cells twice as hard, or even more. Like ideally synergistic, so you combine drug 1 with drug 2, and you get the effect that is equivalent to 3. So this is the setup of the study. What we did is we first screened various different combinations of drugs, then we optimized the platform, in this case mice cells, in which those two drugs that we identified can be optimally co-loaded. And then finally, in multiple mouse tumor models, including a patient-derived xenograft, the one at the bottom, that's actually from a pediatric patient, from a kid. The tumor is basically as a xenograft. It was first transfused with luciferase, so that we can image it when it's in the palms of the mouse. And then we can actually grow it in tumors and see whether this combination protocol works. And what is important here is that this new aspect of combining two different drugs in one nanoparticle, this is something that is really coming up now. So we did a very systematic analysis. We looked at initially almost 900 papers. We found 270 papers that are useful for doing a decent meta-analysis of improved treatment efficacy. And what you actually show is, we found a bunch of interesting notions, not going into details there, but the two key messages is that if you do cancer combination therapy with a nanoparticle, you're always better off than if you do it without a nanoparticle. Right? And then the second thing is co-formulation. That means you have two drugs in one nanoparticle. That's more efficient than if you give two drugs in two nanoparticles. So instead of developing two different mice cells and giving them together, if you put the drugs together in one particle, the likelihood that both of them will end up in the same cell, we calculated it, it's approximately a factor of 12 higher. Right? So it's actually a key strategy now that uses advanced delivery formulations and pharmaceutical technology to improve cancer chemotherapy outcomes. So we did this study, we screened various different drugs. These are 4 different gliomas for ERPG cell lines. We looked at taxanes, tetracyclines, and HDAC inhibitors, and all sorts of combinations. We found that the combination of panobinostat as an HDAC and valrubicin as an anthracycline is synergistic. Right? So 1 plus 1 was in this case more than 4, so they more than doubled their efficacy, both of them. And then we developed a mice cell formulation accordingly. And then the last thing we did is we went to these brain tumor models. This is a little bit inconclusive because these brain tumor models are really difficult to do, so we didn't get statistical significance. But all the mice that were treated with the blood-brain barrier opening with the microbubbles and with the dual drug mice cells, they all responded better than any control group. The difficulty here is with these models, some of the mice they start growing after 6 weeks, some after 6 months. Right? That we need to pool everything because we need to do an MRI on where the tumor is, that we need to focus the ultrasound, we need to prepare the mice cells for each of the mice cohorts per se, basically 1 or 2 days in advance. It's very difficult to controllably do this. The other issue is that some of these tumor models, particularly the one in the brainstem, in mouse, they metastasize. In humans, that doesn't happen. But then they metastasize to the nose, and then the mice die actually not because of the cancer, but because they cannot smell anymore. And once they stop smelling, they stop eating, and then they die because of starvation. Good, so the last 2 slides are not from our lab, but I'd like to share them with you because I think they're very impressive. This shows that this technology can be done on patients safely and efficiently. You see a patient in the top here that is either at 4 hours or on the right side at 48 hours, that within the circle is treated with ultrasound and bubbles, or not, on the left. And then when you look at radioactively labeled trastuzumab, Herceptin antibody, you see that you have a much higher accumulation in the tumor area. This is a brain metastasis from breast cancer than if you don't do this. And what came out 2 weeks ago, and this is really exciting, is that if you do this in glioblastoma, and glioblastoma has very poor treatment options. Basically, temozolomide is the only chemotherapy used for glioblastoma, not because it's a great drug, but because it crosses the blood-brain barrier. Right? And then there's a mutation in MGM2 that makes patients more sensitive for temozolomide under certain situations, and that is then combined with radiotherapy. That is the standard of care. Now, if you give the standard of care first, in the first 8 weeks, you give radiotherapy plus temozolomide, and then you follow up with the normal drug, that's the standard schedule, right? Then you get a median overall survival of 19 months, right? That's the curve in green. If you add to each of those cycles of temozolomide, you add six schedules of microbubble-based blood-brain barrier opening, just 6 times during the course of almost a year. You go to a median overall survival of 31 months. Right? And if you look at this is a 16-month plot, you're almost at 35% 5-year survival in glioblastoma, which is enormous. And the only thing you do is combine a clinical contrast agent with advanced ultrasound technology to open up the BBB to get more drug into the lesion. Right? And this was regardless of this NGM2 mutation, which sensitizes cells to temozolomide. So good, with that, leaving you with the summary, I will not read it out. I'd be happy to take any questions if we still have time, and I'd like to thank you for your attention.
ಬಿಚ್ಬಿಟ್ಟು ಆಂಬಿಯನ್ಸ್ ಸಾಕು.
Yes, are there any questions from the audience? Yes, 1 or 2 questions. Sir, please. Please, sir.
You told 2 drugs in 1 nanoparticle.
Yes.
Can we say that it is functionalized nanoparticle?
It's a normal liposome, so I'm not sure what you mean with functionalized. It does not have any peptide or recognition motif on the surface.
Oh, it's not functionalized.
I think with a peptide or with an antibody on the surface, that is what you define as functionalized.
Yes.
That would typically not perform better. I'd be happy to explain to you in a coffee break why that is the case. Many people believe that if you add a ligand to a liposome, it will go to the tumor better, but it doesn't. Right? It's only step 5 or 6 in the process of circulation, externalization, penetration, distribution, and then it's binding to the receptor. And what often happens is once you have a peptide on a liposome, you will reduce the circulation half-life time. That means the overall influx of the liposomes into the tumor is more than 50% lower. It might stay a little bit longer, or it might go into the cancer cell a little bit better. That's true. Right? But typically, the total amount that you deliver for tumor requirement is lower if you add a ligand. So ligands are, I think that the consensus is that ligands are mostly needed when the drugs themselves cannot enter a cancer cell. And chemotherapies can typically enter a cancer cell. Then it's fine to not use a ligand.
One more question.
Yes, please.
As per current trend, based on biomarker drug therapy is started.
Yes.
Is it possible to, at the same time, all drug therapies are not useful to the same disease of different patients because of variation in the germ, right? With this imaging biomarker identification, is it possible to ensure that all drugs can be used for all cancer patients?
No.
It's same cancer I'm talking.
No, for multiple reasons. So I think imaging it's really too time consuming and too costly to screen every patient with imaging. You do that to see if the therapy works, but you don't do it to select a patient for a certain treatment. That's why we set out to do those biopsy biomarkers. And then the second thing is that so imaging would also only be useful to monitor whether the drug goes to the tumor or to the metastasis. Then you still do not know whether it works. Right? Because it can be molecular sensitivity, you can have development of resistance, you can have drug efflux pumps that will not even allow the particles to accumulate. So I think imaging is useful for many different things, but not to per se help in drug repurposing. I think that's in a sense what you're asking.
Thank you.
Thank you.
ಹಲೋ ಸರ್
Yes, Dr. Boop, yes, Dr. Bhupendra, please.
ಸರ್ ನಮಸ್ತೆ ಸರ್ ಸರ್ ಇದು 82 mm ಯು ವಿ ಫಿಲ್ಟ್ರ್ ಬೇಕಾಗಿತ್ತು ಸಿಗುತ್ತಾ ಸರ್ ಎಷ್ಟಾಗುತ್ತೆ ಸರ್ ಓಕೆ ಸರ್ ಯಾವುದು ಸರ್ ಕಂಪನಿ ಸರಿ ಸರ್ ಹಲೋ ಸರ್
Yeah, it's a great question. I think we can talk about that for 10 minutes, but I'm going to try to do it short. I think we've managed to, not only for these mice, but in general, so in a liposome this will be tougher, but if if you work with polymer particles or other types of formulations, you can work with different types of conjugation chemistry to even entrap drugs that are much likely because of partitioning to actually stay in tumor micelles. We do it with plastic loading, not chemical conjugation. One of the issues is the ratios of one drug to the other. They will also depend on partition. So for doing a ratio, we did a ratio here, I think of 3 to 1. That's easy. If you need 100 to 1, that's much more difficult to control. So that's another issue. What, from a translational point of view, the key issue is that physicians, oncologists, always say we want to be able to balance the dosing of drug A to drug B, which in such fixed formulations is not the case. The funny thing is oncologists do not do that in 99% of cases. Only in very few cases they actually lower one drug because of a specific side effect. So there are a number of hurdles to be considered when taking this forward, but I've seen a couple of ongoing preclinical and early clinical studies that really show added value of this combination in one formulation. So I think for the pharmaceutical technology field, this is something we're going to see more of in the years to come. Thank you.
Any more questions?
I'd like to ask a very basic question, and this is really not my area, so if it's a dumb question, please excuse me.
There are no dumb questions.
We'd always heard a tumor needs good blood supply for nutrients for it to grow. So the whole idea of an actively growing tumor with poor blood flow sounds like a contradiction in terms of a simple matter. So how can we say the tumor is growing aggressively and yet there's poor blood flow?
Because most nutrients that are relevant for a tumor are very small and diffuse very efficiently. So glucose and oxygen have no issue diffusing across, multiple millimeters. That said, these tumors, they do have some blood flow. It's just below the detection threshold of Doppler ultrasound. Right? You're absolutely right with your thinking. There was also this concept of anti-angiogenic therapy. You just knock down all the blood vessels and you would kill the tumor. That didn't work. It only works in combination therapy settings. There is no anti-angiogenic monotherapy that just kills the vessels because tumor A to tumor B, even in the same patient, they respond to different doses, different timings, differently. So what now I think within, at least in part of the clinical oncology field, the consensus is that by doing blood vessel modulation therapy, you stabilize a certain amount of functional blood vessels to help drugs get there better. Only in combination therapy setting. Right? If you would do that with a healthy tumor, well, with an untreated tumor, maybe the tumor would even grow faster. But then again, very short answer to your question. Glucose, other oxygen molecules, etc., they can do a centimeter without blood flow.
Thank you.
Thank you, Doctor.
Thank you.
Trained Lammers, a lovely presentation, very highly specialized, and you tried, you gave us a bird's eye view of how can artificial intelligence be integrated for advancing imaging technology, for improvement in the nanomedicine and their clinical performance.
Hello, sir.
Well, those of scientists who are
welcome to
interested
Media, sir.
in cancer nanomedicine,
हेलो वेलकम टू मिल जाएगा सर वो 82 mm प्रिंटर हाँ मिल जाएगा दो चाहिए कितना चाहिए
in terms of design
सर
of
आज टाइम आपने फाउंडर को
nanomedicines
दिया था दो लिया था
would be
मैंने
based on patient stratification, like say tumor type, as you said, and
फोटो
also
भेजता
tumor
हूँ ये
targeting
77
heterogeneity based on the
अभी
size,
रेट ज़्यादा हुआ
the
है
size of the nanosize, the release
ठीक है सर मैं
characteristics,
आके
and
लेता हूँ
also
शूट पे हूँ मैं खत्म करके
degradation
ले
kinetics.
2 days शूट है संडे आता
Imaging tumor targeting has been
Thank you.
given by you very nicely, and you also talked about mycel tumor targeting with treatment efficacy in terms of high or low accumulation at different places. Well, very good slides on imaging mycel and metastasis targeting I could see. Then to my younger generation, I would tell them that you should look into, if you are working on these type of research studies, look look into AI-enabled imaging technology. There's a lot of literature available on it, and Dr. Lammers has very nicely told us about sono-permeation strategies. It's another very interesting area. He has also told about co-combination of nanoparticles, or as well as nano-combi therapy, very interesting, another technique, as well as nano-sono chemotherapy for brain tumors, for brain targeting. I think there are lovely takeaways for those who will be wanting to work in these highly specialized areas for cancer therapy. Thank you, Dr. Lammers. Lovely.
Thank you, sir, for the inspiring and informative session. Request chairs to felicitate the speaker. Moving on to the third session for the day by Dr. Joanne Chun. Dr. Joanne Chun is the Director, Postgraduate Education Program, Bioengineering and Therapeutic Sciences, School of Pharmacy and Medicine, UCSF, USA. I request
models, and this data feeds into virtual chemical spaces that So to put this simply, industry's demand is outpacing the training. So right now, what we're seeing is many companies restructured their entire R&D departments around data science. So they're looking for the computational scientists, those interdisciplinary trained scientists. But the reality is that traditional PhD and pharmacy programs don't have room in the curriculum to train their students with deep computational skill sets. So here we are seeing that demand doesn't really meet the supply. And that is the gap that UCSF is trying to bridge. We decided to develop a new program that was just launched last year. So we developed a new master's in artificial intelligence and computational drug discovery and development. We call this program the AICD program. So this program is the first program of its kind in the United States that's specifically designed to train scientists across the entire spectrum of drug discovery, drug development, and through pharmacovigilance. And in order to achieve this, we built the program around three core principles. A deep grounding in pharmaceutical sciences, because you need to understand biology, you need to know the questions you are trying to solve. Also, the second principle is a deep grounding in AI and machine learning. So the students are not just learning theory, they are actually learning how to build and deploy these models. And the third principle is the industry partnership. We feel this is really an essential component of our program because it's important for the students to be working on real world problems, right? We truly feel that education should reflect the work that's being done. So this is a high-level overview of what our curriculum looks like. So this program spans 5 quarters, so just a little over a year. And we start with foundations. So the students start learning and take courses in machine learning fundamentals. They also take courses in pharmacokinetics and pharmacodynamics, real-world mining and analysis, and they also learn the modern techniques in drug discovery. And as they progress in the training becomes more technical. So they start learning everything from classical machine learning algorithms to more complex models like deep learning, but it's always applied to biomedical and pharmaceutical questions. And after they go through the coursework, they will spend the last quarter on a capstone project. So even during the coursework, they're already working on hands-on projects. So this is an example, these are just examples of projects that our current students are working on, and this is just within the first 2 quarters of their training. So one project is of FDA chatbot, and this is in collaboration with OpenAI and another faculty in our department who was trained and worked at the FDA. So in this first project, students developed an AI chatbot using LLMs, so large language models, agentic AI tools, and with retrieval augmented generation, so RAG approaches. And the goal of this really was to help scientists interpret and summarize the drivers in AI in the state of California. So it was really a proud moment for our program. So we can see that even within a short time of training, the students are really making great achievements. They placed first in hackathons, ideathons. So these are master students competing with PhD students, with postdoctoral scholars. We have our students also offering peer-reviewed papers, presenting their work at national conferences, but most importantly, they are already securing industry roles. We have students who have secured jobs at different leading companies at A-Star, Genentech, GSK, and Danaher before even graduating. So our program is really preparing students for careers across biotech, pharma, tech, academia, regulatory science, and more. So our students really, when they graduate, they come with machine learning fluency. They have domain-aware modeling and regulatory and ethical foresight, and they can make impact across key disciplines. So this program, I think, if the model really works, it's making impact across multiple levels. Yes, we do non-manual.
For ADMT, non-manual.
We
Yeah.
use SimSip, inline SimSip.
Sim.
Yes, yes.
Thank you so much. Thank you so much. Thank you.
Of course, yes. So we talk to our industry and we use the same tools that they're using. We teach them Win non-Win, SimSip, non-map. So those are the key tools in PKPD modeling. And then you see like in drug discovery in that space, we use AlphaFold, VoltsGen, depending if small molecules, large molecules, the different tools, they're exposed to everything. Any other questions?
Any other questions?
As per your report, it is understood that it is three people are involved, three departments involved. One is industry, regulatory, and your university, right?
Yes.
So, in order to provide good data after testing algorithms and models, they will evaluate for transferability, transparency, and interoperability, and like that, no? Certain characteristics are required for regulatory matter. Who will select these algorithms and models?
So these models, depending on the question, so we have different faculty who work on different problems with different models, and then we actually break up the students into different groups, and different groups of students select different models to see which model performs better for the same data set. So we get to learn different types of models depending on the question. So like support machine,
Actually, who will raise these questions, problem statements? Industry will give?
Yes, industry, they do give us the problems for the capstone projects. And then is a direct relationship only with the student and the company. So when they go to these 3-month capstone projects, the university, we don't look at their data or their models. That's their own proprietary information. And so the students only need to tell us whatever their legal department clears for them to present back.
Okay, thank you.
Sure. Thank you.
Yeah, thank you. Thank you very much, Dr. Joanne Chu. A wonderful presentation again in succession.
Thank you.
And I think this was very relevant to our current teachers, if they are here, and to the students. They should take a lot of inspiration from what Joanne Chun has said in her presentation. It is very important that artificial intelligence is integrated with the various different courses and learning and teaching technology. Well, she has given a very good bird's eye view of the projects developed at their university. And I'm very inspired by her slogan, from hypothesis to healing. Really, it's interesting, I've been listening to various slogans, but this is very good. Well, we have to understand what we are looking ahead. And otherwise, a storm is coming. It's a clarion call to all the teachers, all the students. So I have passed my generation as a student. You have very difficult times, friends who are here. I think, the growth of virtual chemical spaces, virtual clinical trials, even post-marketing pharmacovigilance to be done by AI, moving away from animals. You have seen, we know that, a lot of drug discovery is replaced, we don't need animals for quite some stages. So there are many bottlenecks, as she has said very rightly, and we have more difficult bottlenecks in our country. There's a larger talent gap between the two sides of the find or what do you call it, from the domain expert to the technical mastery. And certainly, I would like to only highlight one thing here, that she has mentioned about the curricular gaps. I think we'll not learn this right away from one lecture or two lectures. There has to be a dedicated learning, and it has to be voluntary. Friends, teacher will not take all the classes. He is not going to teach you each and everything. You have to develop interest in these newer ways, newer technologies, so that you can hasten the process of learning. It's very, very important. I congratulate our expert faculty for having given such a lecture, which is quite relevant to our students and teachers, so that they can change over in due course of time to be aligned with this new technology. Thank you very much. Thank you. Thank you.
Thank you, ma'am, for the remarkable and informative session. I request the chair to hand over the momentum to our speaker, Dr. Joanne. I request all the delegates to scan the QR code and submit the feedback. Requesting all the delegates to scan the QR code given on the screen. Please scan it. Submit your feedback, please. I request our coordinator, Dr. Akhilandeshwari Devarajan, to hand over the memento to our chairperson. I request Dr. Rama Madam also to join us. Request all the delegates to please scan the QR code given on the screen submit your feedback regarding the present session. We are starting with the plenary lecture series 1. I would like to request our Chairperson Dr. V.P. Boone, President IPA, Honorable Professor, Punjab University, to chair the session. I welcome Dr. Gajanan Tharvekar, Director, Acropolis Institute of Pharmaceutical Education and Research Director, Food Testing Laboratory, to chair the session. Requesting the speaker, Dr. Mahendra Patel, Director, Center for Research Equity, Oxford University, London, UK, to give the session. Is Dr. Mahendra Patel here?
Hello. Hello. Is Dr. Mahendra Patel, Mahendra Patel, available over here from UK? Yeah. Dr. M. Raj.
I request our speaker, Dr. M. Rajshekaran, Professor of Urology, Research Pharmacologist, UCSD, San Diego Healthcare System, USA, to give his session. So please.
He's up there. Dr. Rajshekaran. Good afternoon, friends. So unlike the previous session, which was totally dedicated to artificial intelligence, the wisdom,
26.
human intelligence about
26.
artificial intelligence. This session has multi-various interests. The first was probably the speaker is on his way, was on the NHS community pharmacy. The second one is on very novel polymer that is silk fibron hydrogel, which is engineered as drug delivery vehicle. And the third one is a true AI lecture by Dr. Ranjit Varshikar, who is very much here. So we'll have diversity of interest. So let's welcome Dr. Raj Sekhar. He's going to enlighten us on engineered silk fibroin hydrogels. Dr. Raj Sekhar.
Kindly wait. Some technical issues are being solved.
Good afternoon. Can you hear me? Good afternoon. I would like to thank the organizers of IPC for this opportunity to present our ongoing work on testing novel hydrogens for controlled release formulation for sustained antifibrotic therapy. What we will do today is we will present recent progress in our laboratory on the development of advanced biomediators for antifibrotic therapy, focusing on sustained release hydrogen system for the prevention of recurrent fibrosis. Because the fibroproliferative diseases arising from dysregulated wound healing and aberrant tissue remodeling are major contributors of organ failure, account for 45% of global mortality. Current US FDA approved antifibrotic drugs, such as pirfenidone, have limitations due to pharmacokinetic constraints. So, our overall goal is to repurpose an FDA-approved oral antifibrotic agent pirfenidone, which is primarily approved for lung fibrosis, for local application for sustained release to prevent recurrent fibrosis. So we will discuss how our formulation integrates biochemical and biomechanical regulation to inhibit extracellular matrix accumulation, suppress epithelial mesenchymal transition, and normalize cellular mechanotransduction, offering a new next-generation therapeutic strategy for preventing recurrent fibrosis and promoting tissue regeneration. This hydrogel mimics native tissue stiffness and provides sustained enzyme-triggered drug release synchronized with disease progression. So that is the overall summary. Now a little bit of background why research on fibrosis is important. Nearly 45% of deaths around the globe are due to chronic fibroproliferative diseases. Etiology could be inflammatory or metabolic, and it's a fibrosis is outcome of dysregulated tissue repair response to excess deposition of ECM by progressive destruction of normal tissue. So you can see here pretty much every major organ is affected by some type of fibrosis. And our focus is going to be on urethral stricture disease, which is kind of, it is a disease of urethra, fibrotic disease of the urethra. Now let's see the anatomy and clinical burden. Healing by fibrosis places a huge burden on public health. The total economic cost could be in tens of billions of dollars. Dysfunctional healing often causes lifelong disability, which has a significant economic impact. So a little background about the anatomy of male urethra. So, all of us know with a little bit of background in anatomy, urethra is the tube that drains urine from the bladder to the exterior. In the male, the average length is about 22 centimeters. It has got three components: prostatic, membranous, and spongy. So, on the left side, we see the normal schematic of a normal urethra. On the right side, we see urethral stricture, where you see there is a complete blockade of urinary passage, so leading to obstruction of urine. The incidence is not very high, but the problem is it is recurrent. And it is also age related. As you can see in this graph, the prevalence goes up with the age, increase in the age. And etiology could be idiopathic, iatrogenic, inflammatory, traumatic, so forth. And the same thing on the right hand side, the same graph, which shows the age related increase in the disease. The problem is currently there is no FDA approved antifibrotic agent for this disease until 2021. In 2021, FDA approved a drug-coated balloon technology for this condition. It's called OptiLume. It is a dual action mechanical dilation and localized delivery of a chemotherapeutic agent called paclitaxel delivery. Applications for stricture disease and PPH. And mechanism is it inhibits proliferation, preventing scar tissue formation. Minimally invasive, done as an outpatient procedure. So, what are the limitations? This is a chemotherapeutic agent, and it can cause hypersensitivity, and it is not compatible with the implants when the patient has active UTI. Also, it can work only if the length of the stricture is less than 3 centimeters. The other major issue is the cost. The device itself could be about $5,000, and it needs to be repeat procedures every 5 years. So it's very expensive for a patient. For this reason, our research has been focused on identifying novel targets for stricture disease. First, in vivo, we focus on a RNA interference approach because the small interfering RNAs can be designed to target virtually any gene, including traditionally undruggable targets. So we created a stricture model. We use rabbits. We use electrocoagulation method and create urethral injury. Injuries are created in 2 spots about few centimeters apart. And then we evaluated baseline at 14 days, 30 days, and 45 days after the injury and treatment. And we use ureteroscopy, retrograde ureterography, which is an X-ray, and ultrasonography is ultrasound. All these technologies are available in our laboratory for animal use. So again, overall summary, injury and the SIRD, few animals don't get treatment. On the right hand side, few animals get SIRD injection, and we evaluate the same technology. And this particular SIRD blocks transforming growth factor twin and other fibrotic pathways. And the only one side of injured urethra was injected, and the dose was 15 milligram at the injury site. Quick summary of the results of the in vivo data first. So this is the pre-injury. These are the pre-injury images from 2 rabbits, number 21 and 54. You can see the urethra is not obstructed, and whatever you see is imaging dye, and so it is clearly imageable. There is no obstruction. When the animals are not treated at 35 days, you can see that aggravated stricture. There is where you see the arrow, there is a clear obstruction. Nearly complete obstruction at 45 days when the animals are not treated. On the other hand, when you treat the animals, look at the right hand side, number 54, when it is treated with the siRNA, you can clearly see even at 14 days, the passage is clear. So that means it is preventing the formation of strictures or fibrosis. So that is a very good lead for us. We are pursuing on that. Now we move on to the in vitro studies. This is the one on biomaterial-based localized sustained drug delivery, because the advantage of this approach is the drug is already been approved by FDA for lung fibrosis. So this could be, if this is working, this could be easily repurposed without too much of IRB issues. So we tested in 3 different models. In mouse and human fibroblast models, all these are in vitro. Second, in mouse, C2, C12 myogenic cells. Finally, we also developed, current technology, this, urethral and organoids. Organoids are highly recommended by FDA so that the animal testing could be decreased. So we also developed the technology from the patients, from the patient-derived cells. So this is the summary of schematic of structures of cell fibride, agarose, pirfenidone on the right extreme. We have a combination of all, so simple hydrogen bonding, and the pirfenidone. Again, I already spoke about this one. On the right hand side, we have a disease model in vitro 2D and 3D, spheroid and organoids. So in the spheroids, we induce fibrosis with the LPS, and in the organoid model, already they are fibrotic, so we can use them as it is. We use hydrogels to study real-time drug-tissue interactions. Again, this is the hypothesis when the chronic fibrosis model, 3D or spheroids or organoids, when they are exposed to inflammatory agents like LPS and TNF alpha, they change the morphology, the epithelial cells become mesenchymal cells. Now, what are the markers we used? These are the markers we used. We used alpha-sphingosylactin for identifying myofibroblasts indicating fibrosis, cytoskeleton for epithelial cell marker to identify epithelial cells, FOX, stemness and maintenance, DAPI, we know it's a nuclear stain, urocaplin for urothelial cells, and vimentin marker of mesenchymal cells and fibroblasts, and CD44 progenitor or stem cell-like cells and activated fibroblasts. Collagen 1, we know typical marker of fibrosis. CD38 macrophage marker, F-actin myofibroblast marker. Myosin heavy chain is a marker for myogenesis, when it is progressing towards myogenesis. So these are the different markers you will see in our immunofluorescence slides. So as I mentioned, the first approach is testing in mouse and human fibroblast models. So immunostaining summary, it shows that in the fibrotic, the cells exposed to the inflammatory agents, they are fibrotic. They show F-actin and collagen, and also they show fibronectin and smooth muscle actin. And when they are exposed to just pirfenidone alone, there is some improvement. But when the cells were pre-exposed to PFT loaded hydrogel, clearly there is a significant protection against turning into myofibroblasts. And we used Sirius red to stain them. You can see that fibrotic cells are mostly stained in red. In the middle, pirfenidone reduces that staining. Extreme right, the hydrogel offers significant protection, and also it decreases the oxidative stress. One in the green is the PFT loaded hydrogel group. You can see there is a clear decrease in both the level of oxidative stress as well as cell migration, which is an indicator of decreased fibrosis. So next model is C2C12 myogenic cells. This is a mouse cell line, and we wanted to see if this will help us to improve myogenesis. So this is a little complicated co-culture model, that's why we call it 2.5D. What we have done is, in the bottom section, we put the C2C12 cells. On the upper chamber, we put raw cells, which are macrophages. We co-culture them. And also we expose them to LPS. And then, at frequent intervals, we stain them for markers. So when cells that are treated with PFD, and extreme right we see hydrogel. The hydrogel treatment significantly improved myosin heavy chain, which is a marker of myogenesis. What it means is the cells are kind of from the fibrotic pathway, they are diverted into myogenic pathway, which is beneficial. And right below, we saw a graph, bar graph, showing cellular stiffness measured by a technique called atomic force microscopy. And we see that the fibrotic cells have a significantly higher stiffness, whereas with the treatment with hydrogel, the green bar, there is a significant reduction in the stiffness. And also the bioenergetics we measured, the red one is the myofibrotic cells. There is a decrease in the bioenergetic production, which shows us there is a mitochondrial dysfunction, which is prevented by the hydrogel treatment in green. Next, we also did, what is happening at the mitochondrial level using EM. The one on the left is the control, the middle one is myofibrotic, the right one is the treatment group with the hydrogel. You can see that compared to control, the myofibrotic group, there is increased swelling and overall enlargement of the mitochondria, whereas that is significantly protected. This disarrangement or swelling, etc., is significantly protected by the treatment group in the right extreme, which shows that even at the ultrastructural level, we can see the protection by the treatment with this hydrogel. Finally, we went on to organize. We get tissues from the patients when they undergo a surgery called erythroplasty, and we can culture them to make them patient-derived organoids. Organoids, people who are working in the field, they will know that it is highly preferable to 2D cell culture model because it retains a lot of native properties of the disease. Highly recommended for drug discovery now. What are organoids? These are 3D structures derived from stem cells that recapitulate tissue architecture and function, replicate complex organization and cellular heterogeneity of native organs, and more physiologically relevant than conventional 2D cell culture. Can be generated from patient-derived tissue for personalized medicine. So definitely, as I mentioned, it's got a lot of advantages. So applications, it could be used for disease modeling, drug screening, personalized medicine, safety evaluation. And this is now recommended by both FDA as well as for National Institute of Health for drug grant applications. This is a recent article in 2023. You can see on the right what are the main applications of organoids as a disease model, as a drug discovery and toxicity assessment, personalized medicine, and regenerative medicine. In all those areas, these organoids are replacing the animal models. So what did it establish? We get tissues from the patients, tissue fragments are enzymatically digested, and then growth conditions are optimized, we characterize them, and finally we validate them. Spheroids treated with the LPS and the TNF alpha, they show increased vitality and collagen, whereas the cells that are treated with the peripheral, they don't show those markers, and the marker expression is much more increased with the hydrogel compared to just peripheral. And this
these are all summarized in the 5-day treatment on the right in the bar graphs. So overall, pirfenidone-loaded hydrogel treatment effectively suppressed these pro-fibrotic responses through sustained release kinetics and ECM limiting properties. And also we have shown on the left side is the tissue stain for the markers, the whole tissue. You can see that it expresses collagen, TGF, both are the markers for fibrosis. And on the right hand side, on the top, it is control tissue. You see some collagen, but you don't see TGF, etc. But in the patient-derived organoids, you see all the characteristic features of the whole tissue. You know, as I said, patient-derived organoids represent, the whole tissue, and we can manipulate a lot of things in the in vitro system. So, in conclusion, this is the first integration of spheroid and patient-derived. To the best of my knowledge, our lab is the first one to develop this organoid model for urethral sphincter disease, and we are testing it with this hydrogel. This platform uniquely bridges preclinical mechanistic discovery with the clinical translation by combining patient-derived disease models with the therapeutically deliverable biometric system, enabling mechanistic insights to directly inform localized treatment strategies. And you can see that, it shows urocapillin, which is a urothelial marker, and some amount of cytokeratin, vimentin, everything can be seen in this. Finally, we would like to thank all the contributors. This work was primarily done by one of my graduate students from IIT Madras. She has been with us for the past 2 years. She is finishing a PhD, and others are all clinical collaborators. Thank you for your attention. I would be happy to take any questions.
Thank you, sir, for the enlightening session.
Thank you.
It's open for queries. Any questions?
By the way, this is not a painting, this is a beautiful picture from our San Diego beach, and it was captured by one of my close friends in the audience, Professor Ravindran.
Thank you so much, Dr. Rajraj, for enlightening one of the less explored features of silk fibron hydrogel. So this has been explored extensively also, but you have you have reiterated its use in not only controlled release applications, in wound healing and many other purposes, and with extended release, but you developed spheroids, organoids, which is way high, in the technology, which is needed and which is recommended by the federal agencies, like you told, US FDA is very, vociferously these days recommending the use of organoids, to reduce the animal sacrifices, very nicely put forward, and these silk fibroin, polymers, these have very characteristic properties, being bioresponsive, sold gel formation. It is a very, very characteristic feature, and they are greatly structured, used for tissue engineering as well as drug delivery purposes, biocompatibility, biodegradability. These have been the features. We at Punjab University have also used, but though that work was relatively primitive in contrast to the work which you carried out. I must compliment you, for such a high degree such high quality of the work carried out at University of California and San Diego under your leadership. And the house is open for discussion, please. We can have one or two questions, from the audience. Very innovative work, very useful work, and it can be used for not only fibrosis, the disease which he has highlighted, wound healing, many other, diseases, very successfully. Yes. If there are no more questions, let's compliment and give him a very big hand, Dr. Rajdev. Thank you
for the response.
Requesting the chair to felicitate our speaker with the momentum. I request our next speaker, Dr. Ranjan Barshikar, CEO, QVD International India. He had a long career in pharmaceutical industry with specialization in QA management, AI, R&D, regulatory, and has specialized in quality by design. Request you, sir, to please start the session.
Before he starts, let me compliment Dr. Ranjit Barshikar ji. He's a very senior scientist, who has more than 4 decades of experience in industry. I know him and I know his work. And I think five decades. 5 decades. It doesn't look like to have 5 decades of experience. The most remarkable feature in the persona of Dr. Ranjit Barshikar is for everybody of us to learn his adaptability. When he learned there was no QBT, there was no AI, there was no computational pharmaceutics, but he is moving ahead of the younger generation in learning those technology, adopting those technology, and making himself proficient to such an extent that he now trains people, and teaches the younger colleagues on these technologies. Welcome, Dr. Ranjit Barshikar ji. He is going to enlighten us on AI applications in pharmaceutical industry, particularly industry.
Thank you very much, Doctor, for the nice introduction. Appreciate very much. Congratulations to you for an excellent award you got today for your tremendous contribution to the education and the industry. And you are still continuing in the advisory role for Punjab University. Yes. Thank you very much. Fortunately, I am able to get 5-10 minutes extra today because one will speak is absent. So I am going to use that time, doctor, if you permit. Or 30 minutes? No problem. Don't worry. Sure. Sure. No problem. No problem. So, good evening everybody. And I'm going to focus my talk on the AI application in pharma industry. How it is being used today. And how many companies are doing it. Indian companies, generic companies in Europe as well as in US, branded organizations who have patented the various drugs, the multinational companies, and also I am going to focus a little bit on future. Though it is a future for all of us, but it is a reality of two companies in the world. Absolutely, fully 100% paperless factories, digitalized plant, minimum human intervention. So I will show you one video on that by the end of my presentation. It is about 3 minutes. It won't take much time. So that will give you a very good idea what is happening in pharma world today and what students should pick up from that perspective. Okay. Regulatory agencies, whether it is US FDA or European agencies, they have gone much ahead in guiding the industry from AI perspective. They have the documentation being issued initially by US FDA in various forms and by Europe this year they have issued AI Act for the industry and for themselves as well. And in January 25, US FDA has issued this document as a guidance for regulatory decision making for drugs and biological products, guidance for industry. So this document also speaks very well about the use of AI and what are the applications they expect regulatory agencies while using on or manufacturing our products, testing our products, releasing our products, market survey of our products. Okay. So AI, as you will see on the screen now, it is being used today in every department of pharmaceutical company. All the departments, right from drug discovery to R&D, quality, pharmacovigilance, manufacturing, marketing, and obviously clinical trial. We have discussed that in last 2-3 presentations about clinical trials as well. And there are lots of more data. One simple example I will give you what is happening in the industry. Look at this, drug discovery. Drug design, drug screening, drug repurposing, drug structure, and drug screening from thousands and thousands of molecules, which is going to be successful drug research, which is likely to be a blockbuster. That also can be used by this AI technology, and it is a reality today. Similarly, which are the different type of suitables required for experiments in finished product, dosage form, monitoring and the development of the product fast. Now QBD, it was there for more than 2 decades now. Now slowly QBD is being taken over by AI, especially in design space area. So modifying development, in-process specifications, control procedures, predictive quality decisions, how it should be to avoid the problems.
వాళ్ళకన్నీ స్కోర్
And like that, there are so many other advantages for every department in pharma industry.
ಶೂಟರ್ ಇದ್ದೀರಾ
In drug discovery,
ಓ ಕರೆಕ್ಟ್ ಹೇಳಿದ್ರಲ್ಲ
you will
ನಾನು
see
ಕರೆಕ್ಟು ಇವಾಗ ಅಡ್ರೆಸ್ ಎಲ್ಲ ಚೆಕ್ ಮಾಡಿದ್ದೀನಿ
drug design and drug screening, to be precise, these two more focused applications for prediction of the toxicity. And I was talking to afternoon some of the experts here during the morning program. The toxicity now, slowly in near future, all animal testing will go away, and it will be taken over by AI, by predictive toxicity studies. And the discussions are on with US FDA in this respect by Indian companies. Already submitted the data to them, and how the predicted toxicity matches with the traditional toxicity studies by using animals. So that is going to be a big revolution in the research area in time to come.
ಕಳಿಸಿದ್ರಿ
Similarly, polypharmacology, where drug acting at multiple targets, repurposing, predicting 3D structure, and virtual screening of large number of compounds to evaluate the structure of novel compounds. AI, new antibiotic has been developed now by the scientists. The MIT scientists in US has developed a antibiotic called halicin. Halicin. They have named it as halicin. And interesting part of AI, using AI to develop the antibiotics, is that it is not showing any resistance from the body to use. That's the biggest advantage. In recent times, several antibiotics which come to the market, within 1 or 2 years, the humans start showing resistance. It becomes ineffective. Whereas AI designed antibiotics are not having any body resistance. One example which is here is with the AI, there is no resistance, and this is ciprofloxacin showing the resistance in the body. And currently 50 plus antibiotics have been developed by the scientists, and they are in various clinical phase studies. Similarly, FDA has received 500 plus submissions where AI technique has been used. So you see how much the world is going ahead, and the more our students learn on this kind of techniques, it is going to be beneficial for them when they enter the industry or when they start their own activity. This is the interesting part. Now, in this molecule developed by in silico medicine, and this is now first molecule in the world which has received the generic name, which is called Rentosatin by United States Adopted Name Council, USAN, and this is end-to-end AI platform has been used by them, and this is first where both the target and compound were discovered and designed by generic AI. Generative AI, we call it Gen AI. And at the moment it has gone into the clinical phase 3 just 2 months ago. And it has got very positive results in first 2 clinical phases, clinical A of 2A and 2B both. And the discovery time, surprisingly, is less than 18 months, which normally takes around 6 to 8 years. And the cost of this development within 18 months is around 10% of conventional or traditional way of designing the molecule. You see advantage of the speed and the cost. Okay. So in silico medicine is the first company which has come out with this kind of approach, which is in now third phase. This is a graphic which is available as on today till 23. Afterwards, there is no data available. But up to 23, 172 such molecules have been developed in various stages, like AI discovered targets, AI designed small molecules, antibodies, vaccines, repurposed molecules, and etc. others. So every year there is see the progress of development of the molecules from AI perspective. In clinical trial, you can see the predictive analytics, advanced predictive analytics, faster identifying the candidates now. This is a further advancement on the clinical trial area. And remote monitoring and real-time data monitoring activity or the real-time data access for a high security, using e-records to reduce the data errors to enhance the efficiency of the development, thus reducing the clinical trial time by almost 50%, and reading the volumes. Now normal any book, 300-400 pages book, we take several hours if we sit continuously. For some people, several weeks. Whereas AI takes in seconds, it reads the whole book in seconds. An interesting part, the AI doesn't forget. We can forget, AI doesn't forget. And obviously the cost saving and the speed. In QC lab, complete documentation check, document digitization, data analysis, and the important part is anomalies identification in testing. The same thing also happens in manufacturing. Any issues or any bottlenecks are likely to happen something wrong, AI tells in advance before the error happens. And AI also informs to the relevant people that how to solve the problem. And it gives 3-4 suggestions. Also it gives the detail of which is the best option to solve the problem so that there is no downtime. There is no failure of the process. So you save the batch. And the consistency in manufacturing goes on improvement. Nitrosamine, which is being talked today in last few years in a big way, repetitive jobs, it takes over complete repetitive jobs, and analytical method development also it guides, like product development. So which is also additional benefit to the organization. You see documentation, change, digitization, then most important is
ಕೋರ್ಮಂಗ್ಲಲ್ಲೇ ಇರ್ತಾರೆ
data integrity.
ನಾನು ಅದೇ ಪ್ರೈಸ್
The whole
ಹಾಕಿ
industry
ಸಮಥಿಂಗ್ ಬರ್ತಾ ಇದೆ
has gone through a very tough time since last almost 15 years
ಅಲ್ಲ ನಾನು 8
because of the data integrity issues.
ಕೋರ್ಮಂಗ್ಲಲ್ಲಿ
Most of it
ಅದಕ್ಕೋಸ್ಕರ
were unintentional.
ಅವ್ರು ಇರುವಾಗ ಬುಕ್
But
ಮಾಡಿ
still,
ಅಂದ್ಕೊಂಡೆ
these companies in the
ಅಲ್ಲ
world
8 o'clock
received
ತನಕ
the
ಅವ್ರು
warning
ಕೋರ್ಮಂಗ್ಲಲ್ಲಿ
letters
ಇರ್ತಾರೆ
of
ಸೊ ನಿಮ್ಮ ಮನೆಯಿಂದ
non-compliance, which is a major blow to the companies for suffering their billions of dollar business world over.
ಹಾಂ 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 ಹಾಂ
So this AI AI is tremendously helping now monitoring
ನಾನು ಬುಕ್ ಮಾಡಿದ್ರೆ ನಿಮ್ಮ ಮನೇಲಿ
and reviewing the data which is coming out from every equipment to ensure that no error is happening in documentation. If any blocks are filled by AI, it again wants to the relevant people in the company to take care before the error happens. Remember, okay? So data integrity and compliance is the biggest advantage now to the world over industry. In analytical area, during and post-analytical tasks, the AI is being used in a big way. As I told you, audit trail reviews. Audit trail reviews normally takes to the quality assurance team to check one batch document for 2 to 3 days minimum. And you see every day several batches are being manufactured by the company. So it is important that every document should go through quickly for review mechanism by QA, and even though it is done by QA, likely errors because of human checking it. But advantage with AI that it reviews in next second. As soon as the data is generated, it reviews, it gives its comments. See the advantage of time again and accuracy of information. So complete data integrity compliance is the biggest advantage to the industry. In quality management system, we check that fully automated jobs right from SOP generation in the industry. Adequacy of SOP is important. So AI does this job in seconds, guides the company how to do it. So there are several smart quality perspective. We see AI-enabled quality solutions which are already available in India. An interesting part that I am very proud of our Indian companies as well, who are designing the software for AI. We are very much ahead than the world. Our India is an IT hub, and that's a plus point to our pharma industry as well, to go ahead in a big way for AI implementation. So we are moving towards the proactive quality, internal auditing processes, planning for audits, review of the audit reports, generating the communication for auditing purposes. So it has a significant cost saving, productivity improvement throughout the organization, and quality monitoring process in the operations. And this is very, very important, my friends. The predictive analytics, predictive maintenance, these are the two very important advantages because of AI. I explained to you predictive analytics already. Predictive maintenance is the system where AI guides the organization that which equipment in your factory has a risk of failure and when in advance, 4 to 6 weeks before it fails, and it informs to the concerned people to take corrective measures so that equipment does not fail and your downtime of manufacturing reduces, and there is a consistency in manufacturing, that failure is also avoided. So predictive maintenance plays a major role in compliance. In packaging area, robotics is now being used in a large sections in sterile injectables, packaging or filling, in quality control testing also, accurate quantity to be used during testing. So we are moving towards smarter, safer, sustainable, even in secondary and tertiary packaging. These robotic hands you see here, that checks the label 100% correctly, whether everything is in order, whether there is any mix-up in packaging. So we can easily avoid the mix-up of the products. There are several Indian companies, global companies I have been using it in India. Sun, Cipla, DRL, Lupin, Arvindo, Piramal, and there are several more. At least 100 companies have started using this AI technology in a phased manner. And Sun Pharma has gone much ahead by starting their own AI company in 2011. And where they have come out a strategy of AI implementation to predict the toxicity of the products, to eliminate animal studies. So I don't have hesitation on naming that company because everything is in public domain. And they all are in phased manner. Drug discovery to supply chain for quality monitoring, and the robotic process automation, which we call RPA tool for data integrity compliance, which I have installed in one of the companies in Hyderabad, and that is being used for last 3 years successfully. And paperless plant. Real-time verification. Now validation is one of the areas which is very important. AI has taken over completely validation process. All types of validation it takes here to move towards the next generation of validation system to avoid any problems. Supply chain, blockchain, you know the purpose of the blockchain is more safe information. Like we construct the wall in the reality industry, the walls, bricks are connected to each other. So each brick of pharma has a different information and it cannot be easily hacked. Tomorrow if something comes out extraordinary, we don't know. But it is a wonderful technology. So far it has been safe and you can trace to the last user of your batch in the market. To identify fake drug also, we can use AI technology to identify the fake drug. And the interesting is the case study is true tag technology, which is being used now, which is using AI powered micro tags and blockchain to ensure traceability and authenticity of the drug. So it is not easy to copy the product in the market. And you can make out if somebody is complaining about spurious drug, you can test that with the help of AI intelligence. This is one which has got HoloLens, Microsoft HoloLens. There are so many companies which have come out in the world today by having this technology. This HoloLens is now being used to see, with the help also of twin technology, virtually see your equipment, virtually see what is happening in your machine, so that you can easily find out what is wrong inside the machine or everything is going smoothly inside the machine. And the technology here is that when you use this technology, something is wrong and you need expert help, whether the supplier expert or the company's expert somewhere who knows this equipment, but at a longer distance, use this HoloLens. That expert also uses HoloLens and virtually your equipment problem and on the spot guys what to do. So it saves time. Downtime is reduced again. And this is also being used for training purposes now. I have several videos. One such video is the person is talking in LA to the LA audience, and the second audience is in Japan, Tokyo. But the original audience speaker doesn't know Japanese language, but virtually present there with the help of hologram. The same outfit of the person and talks in Japanese language, but she doesn't know Japanese language. See, the advantage is also to be used in training purpose. From design, manufacturing to sales and operation, everywhere, mixed reality, augmented reality, virtual reality, everywhere this technology. This is what virtual screen they generate while using this technology of HoloLens. Robotic, automated change. Also in manufacturing of sterile, I'm sure many of you know that we need minimum 4 hours the media plates to monitor the environment to be kept inside the sterile. Now this automation is helping to change the plates automatically. You don't have to touch. So you are again minimizing human intervention in sterile manufacturing. This is a Pfizer's plant. This is now, this year it has been operationalized, and with the help of technology which is called modular aseptic processing. And their whole plant, they are using AR, VR, MR in different activities in addition to the training to reduce the human error, highly digital, highly robotic, digital twins and predictive maintenance is fully used. Minimum people working in the plant, core areas, okay? I am running the video here. You see for us it is the future, but for one company called Sanofi, it is actually they have started using this plant. Can you run the video?
With increasing advances in medical science and innovation, complex medicines are set to become even more important in improving people's health worldwide. To keep pace with this shift, Sanofi is committed to ensuring our industrial capabilities are as innovative as the drugs in our pipeline. To enable this transformation, a €4.7 billion investment program has been underway over the last 5 years. The factory of the future will be digitally enabled. In the evolution of our plants, we will see the adoption of new connected and intelligent equipment, more sophisticated process technology, and solutions to augment operators with additional capabilities. It will allow us to deploy advanced data analysis solutions to further improve the quality, yields, and reliability of our facilities and optimize our inventories and logistic flow. This plant will produce monoclonal antibodies in single-use fermenters. All production and setup steps data will be documented in real time, resulting in a paperless manufacturing solution with electronic batch records, review and right-time release, and real-time process control. Leveraging 3D models from engineering and real-time data during the production process, it will be possible to optimize operations by using a digital twin, mirroring the real plant and enabling simulations of changes. Digital twin technology will also be used to train and prepare operators before the plant has been built. The first stage of the production starts with the the upstream process, fermentation and cell amplification. For this, we use single-use bioreactors with disposable bags instead of a culture vessel made from stainless steel. Single-use bioreactors will change the way we operate and organize our facilities with a higher level of flexibility. All bioreactors are equipped with inline sensors to allow multiple measurements of various data and parameters. It creates a fingerprint of the process. Therefore, all procedural irregularities are spotted immediately and rectified to avoid any impact on the product's quality. Once the cells have been produced, the next step is to purify the drug. Again, all the necessary pieces of equipment will be closely monitored by a series of sensors that share information. A standard batch would thus create more than 3 billion data points from over 5,000 parameters for every single batch produced. That's a lot of data. It will all be transported by a reliable and high-speed network and a sophisticated storage and analytics infrastructure. Once the data has been analyzed, employees will be able to access its findings via a number of interactive screens located throughout the plant. Continual monitoring of data allows for a better understanding of the complexities of the production process. The ambition for Sanofi is to create a fully fledged and disruptive biotechnology sector, answering the demand for customized and high-quality products. A patient-oriented, flexible, digital and sustainable ecosystem. This is Sanofi's vision for its factory of the future.
Such a fast change, revolution is happening in pharma industry. And imagine what will happen after 10 years. Absolutely fantastic. Changes are happening, unbelievable changes are happening. And all is due to AI. So, in manufacturing, it is avoiding the deviation, real-time monitoring control, the effective use of the operational equipment goes up by 50% in the company. And all others we have already discussed. So, the timely anomaly detection, predictive maintenance, predictive quality, shaping new era of excellence in pharma industry, my friends. Thank you very much. Any questions?
Thank you, sir. Wonderful information. But I wanted to know that regarding predictive toxicity, how much information are sources available for academics? Because as you know, as I tried with web server, whatever it is predicting, I don't know what is the background data, how much they are using. But I know that data to the products, 60 chemical molecules, building blocks are available in corporates, and that is not open to the academics or open source. Then how much we can use?
It's interesting, huh? So I appreciate the question. Really difficult for me to really answer that. What is how to use it? But really, you see, the complete search you do. Okay, there are so many available social medias are there today, public domain information, as well as please visit the top organizations' websites. They are themselves giving these details. Certain portion they always be held from the public scrutiny, but whatever is required for us, what we expect is already there in public domain. For example, Pfizer or Sanofi or Sun Pharma, it is there on the website, the detailed information. And for example, Sun Pharma's IT head was talking to one of the press chiefs. That information is also available. And I have picked up the information like this. And there are several publications also in journals. You can always access those. Every day I am getting more than 50 such blogs and the journals which are free of cost. Free of cost, imagine. And there are several courses also available today, free of cost, for students to learn from the basic perspective. For advanced, the very minimal charges are there, and that also in India it is available in Indian rupees, and those are the best of the institutes in the world. So we have to access, spend time on that, to make your own folders. I have thousands of folders on AI only. I am ready. And whenever required, depending on the subject given to me to talk, I prepare my presentation based on that, giving that reference. Credit should be given to the concerned people.
Really, really commendable, for since so much of databases, because AI is based on data, the novelty and the accuracy of people, where the novelty as well as accuracy.
Did I answer you?
And okay. But one thing I must add, said US FDA, sir, you must be knowing, in 2022, soon after COVID, 22, they allowed the animal toxicity studies, during preclinical studies, to be replaced with lab on a chip, organ on a chip kind of experiment. You must be knowing. Those are microfluidics. And at that point in time, I still remember emulate software, which was partly AI based and majorly microfluidics based. That equipment, if you demonstrate that the liver on a chip, the brain on a chip, the kidney on a chip experiment, gives no toxicity to that drug. So, the FDA used to grant go ahead. If emulate software tells that it's not toxic, it's not toxic.
Absolutely. Absolutely.
So it's an amalgamation of AI as well as lab on a chip, microfluidics kind of experiment. So AI tends to supplement and complement such type of disruptive technologies. Similarly, 3D printing, it acts synergistically with 3D printing in the design of so many, as Sir has pointed out, design of so many, equipments and so many of combinations, even tablets, for the pediatric and geriatric use. So lot of scope, lot of future is, still needed to be unearthed. So with the help of thank you very much.
Anyone else? Yes.
So thank you for an excellent talk. You gave a lot of detail, very positive outlook, which I think is justified. But could you talk for maybe 2 minutes about the errors in AI?
About?
Mistakes. When it makes errors, when it gives you wrong data.
Oh.
It does sometimes. So if you could just give us a little inkling, I think that would be good. Thank you.
You want true answer from me? I don't know in reality, but industry may vary, some of the companies, that everything will will come to the surface. So far they were managing. Because everything will be captured by here. Everything will be on the screen. And you cannot hide anymore. It's a humongous job they are doing, and in reality, AI is reshaping the industry. Reshaping. Yeah. And lot of people are afraid of that it is expensive, what will happen. No. Those kind of challenges are always there for any new things in the world. So if I want to implement, I can find the sources. I can find the resources. There are several startup companies in India who are supporting this at a very low cost. So they are doing wonderful job. Yes.
I'm fully on board with AI. I'm not knocking it, and I use it a little bit. I'm down there, but I use it a little bit. And I know people in other industries who are using it, and from time to time there is some error. It gives you a nonsensical answer. It doesn't fit at all. So there are those issues, and I think just for balance, I was trying to say, this is so great, but let us just keep in mind there are some issues where it doesn't give you a sensible answer.
Over the years. So this is a new thing, right? There will be always a teething trouble. Always. So it will get sorted out. It's a question of time. Yeah, and everybody will start using. Now today you see the smartphone. What is smartphone? It is AI. When you handle application of Amazon and go through some items like say ladies dresses, saris, you see one example, then next few days you will continue to get several supplies of that information to you automatically. What can we say? So we are so much used to it now. Earlier when we were using the dial phone number, we were remembering the numbers of so many people. Today we don't remember a single person's numbers. So we are using it so much.
Sir, thank you very much for your enlightenment. summarize, so but please pardon me because I have to summarize lecture of a person whose total experience, professional experience is more than my age. So really, myself along with those who are present here will be really fortunate to listen to you. And you have actually given us all the insight what the AI is doing in the industry. And we were surprised to know that already 50 antibiotics are already in the, have been developed and many more are to be introduced. And earlier we used to say that the drug discovery, because of its long process and very high cost and very big infrastructure it was needed. So we used to tell that it is not a cup of tea for the even Indian multinational also. But you have given the example that many Indian industries have already invested and they are running ahead. And that is that again you have explained the role of AI in regulatory as well as this because every some other day we used to read the one this such and such company have received a warning letter 483 and all this because of data integrity and all that. So that problem has I think it will be reduced or it will go to the because much of time and energy will be invested by the company once it gets this. So thank you very much, sir, for giving us this nice insight on the India.
Thank you, sir, for the wonderful session. It was quite enlightening. I request our chair to felicitate. Requesting our chair to felicitate our speaker with a momentum. requesting all the delegates to scan the QR code given on the screen and submit your feedback. Request all the delegates to stay back.
All are requested to give the feedback, please. We will move back to the next session soon. Delegates are requested to give the feedback, please. We are moving to the third session, Micro Lab Sponsor Symposium 2. For this session, I am delighted to invite Chair of Session, Dr. Swati Srivastava, Deputy Director at Ministry of Health and Family Welfare. We welcome you, ma'am. Dr. Swati, having about 23 years of experience in regulatory field and in biological inspection, we are very blessed today here, ma'am. So we are starting the first session. Dr. Harikrishna Maran, sir. I welcome you, sir, for the first session. Sir is Chairman of Vision Digital Media. Sir is giving the topic on future of AI, scope and opportunities of AI in education and industry. Over to you, sir.
Thanks to Indian Pharmaceutical Congress team and inviting me here to share about AI, what exactly the future of AI, how AI is going to take our lives and how AI is going to change the way we live. Thanks to the chairman. So it is wonderful to be at Indian Pharma Congress here. I still remember as a student, my first IPC was in 1991. I was just talking with a older person. So in 1991, we had an Indian Pharma Congress which happened in Delhi at IIT, where my topic, the future of AI. I think most of you people was reading and going through a lot of information on what exactly happening on artificial intelligence. So some of the great things which are happening, and from morning I think a couple of speakers were talking about AI. My previous speaker was talking about how in pharma industry AI is integrated in drug discovery and new innovative ways to make process faster. So my talk is more next 20-22 minutes. I'm going to touch more on how AI is going to be in the next 10 years and what it is going to add to the wealth of people, wealth of companies, wealth of nations. So that is what my talk. And so we run an AI company, which is one of the India's leading AI company, which started 11 years back. It's called Vision Digital India. So I am going to talk what exactly we do on AI also. So AI is not just a future, it's India's opportunity. The reason is, in the last 10 days, I think you might have read in the newspapers, the kind of investments which are coming into AI. One of the biggest investment of Microsoft, which is close to around $16 billion. Then the last month, Google already pledged their investment to set up world's largest data center in Visakhapatnam with $20 billion. Then the next biggest investment of Amazon, which is $35 billion, which they are going to invest in Bengaluru and Mumbai to set up AI-related activities. Then just few months back, the Open AI, which all you people and across the world people use, the ChatGPT, they started their second office after US in Delhi. And most of you people are familiar now what Meta is doing, what Apple into big time into AI. So all together in the last 30 days, India got investments closely 95 billion. 95 billion investments coming to India only on AI. So before coming here, I was just checking what is the total size of pharma industry, Indian pharma industry. So they mentioned it is $50 to $60 billion. So just imagine with the 1.45 billion people in India, where we have to use the medications and medicines. So which is just 50 to 60 billion. In the last 1 month, on AI, we are getting 95 billion investment coming. So that is the scope of AI. That is how every industry, AI is going to create large number of opportunities. And most of the times we read in newspapers that AI is going to replace jobs, we are going to lose the jobs. So how many people will agree for that? AI is going to take over our jobs. Do you all agree? Or you have other version? So what I'm trying to say with a lot of observations, AI is not going to take over the jobs. AI is going to create more number of entrepreneurs. So that is what the Elon Musk, the richest man in this world, just weeks back, he said, the number of millionaires or billionaires created in this world in the last 3 decades. In the next 1 decade, AI will create the trillionaires. And that is coming to reality because there is no trillionaire till now in this world. So now, I think you might have read just 2 days back in newspaper, the Elon Musk company, apart from the Tesla, SpaceX, this is into the space research, is going to list in US NASDAQ stock market. So the valuation of this company is valued at 700 billion. 700 billion. And the Elon Musk, as a major shareholder, his wealth increased to 165 billion he added 165 billion to his wealth. Now, the second richest person in this world and the first richest person, Elon Musk, the difference is almost more than double. So, why I am giving all these figures, why I am telling all this, is today the world is moving towards AI. So, we are Vision Digital India. What exactly we do? I want to bring a practical, what exactly we are doing from our side. So this is on a mission, this is called AI Training Mission, which we just launched 5 months back. This was, we developed from an idea from one of the students actually. So one of the students came back and told, the AI teaching in a classroom happening like a regular other subjects. It is not going on through a practical way. So we said, why not we put some head on, do something on this. So this student went to Intel to do his internship, the 6 months internship. And there, this product was developed, and this is a first AI training mobile mission, where you can carry this mission into the classroom, whether it is a college, whether it is a university, whether it is a pharmacy, management, engineering, whatever subject. You can give a practical exposure of AI in a simple way in a classroom. So that is what this mission. So it is called the Mobile AI Training Mission. So this mission, because now most of you people might have reading again in the last 2-3 days, India is ranking number 3 in AI technology skill manpower. That means in the world where we are a 4th largest nation with a 4 trillion economy, India is number 3 in AI. If you include the people who are working in US, the Indian origin technology people, and combine that, we are number 2 in this world in AI. So that is the kind of a scope what we have as a pharmacy people, pharmacy professional. You may be thinking, what exactly, how it is going to change my life, how it is going to help my career. So not only in pharmacy, when it comes to even legal profession, when it comes to medical doctors, when it comes to the writers who create books, who publish books, everyone now will have a great opportunity if they can able to utilize AI in a better way. So that's how we named this mission after, we call this mission as a MODI. So MODI, the name is Mission of Digital India, because our organization name is the Vision Digital India. So on his birthday, we gifted to him as a MODI mission of Vision Digital India. Then, what exactly we are doing when we are talking about AI, when we are talking about so much wealth it is created, and so much awareness AI is getting across the world. So we thought, why not we come with the new ideas and implement some new initiatives. That's how we brought, when food is served in the train, why not AI is on wheels? So that is the idea we have taken, and now we created AI on wheels. The first vehicle was launched in Maha University in Manipur. And it is going around in Udupi, Kundapura, Bengaluru, all these schools, the government schools, and they are educating on AI using this practical AI training mission in rural schools where the students are not at all familiar about what exactly artificial intelligence. So in the last 4 months, which we developed this vehicle, is visited almost 95 to 100 schools and educated around 25 to 30,000 students. And our vision in the next 5 years to reach at least 1 million students adopting this concept and launching these vehicles across India. So already the work is started now. This mission doesn't require internet. Now we are trying to adopt, creating a solar panels where the power is generated, where you don't require electricity to run this mission. So all these new innovations are going, and in Delhi we already started trying the CNG gas vehicles to move around and educate the kids in UP mainly and NCR area, in Greater Noida, in Gurugram, in Faridabad. So this is how AI education, if we can able to start in early, I think India will become a number one in this world, sure. So the reason is, unlike any country in this world, the greatest advantage what we have in India is our demographic dividend, the youth. So out of 1.45 billion people, we have a 60% of our population is below 35. So that is the advantage. When we give us skills of AI, then definitely our economy will go up, and what we are talking of Viksit Bharat, of reaching number one country in this world by 2047, when we celebrate 100 years of independence, definitely we will become number one in technology, in wealth, in every space of this world. So that is the mission of Vision Digital India, where we are trying to educate people, providing AI skills to all. So then from there we are not stopped, because we thought when we are able to launch AI mission, why not semiconductor? Because most of you people are familiar and might have going through some information on semiconductor. We use highest number of the chips, the semiconductor chips in the world, compared than any country in this world, because we use all our mobiles are connected with the semiconductor chip. All our electronic devices, whether it is a TV, whether it is a fridge, whether it is a car, everything is connected with the semiconductor chips. But we don't have a knowledge on semiconductor, we don't have a kind of skills on semiconductor. So we thought, why not we introduce the semiconductor training mission. So that's how the journey started with the semiconductor. So now, when it comes to the semiconductor, again, I think you might have reading information on NVIDIA. NVIDIA is a very new company when it compared to Microsoft, when you compare with IBM, because IBM is one of the world's oldest technology company. They are the people who brought the personal computer to this world, which is 108-year-old IBM. Microsoft is again more than 50-year-old company, but NVIDIA is not that much old. But today, when it comes to the market capitalization, they are a $4 trillion company, which is equivalent to the Indian GDP, NVIDIA. What they do, only the chip manufacturing. So the biggest opportunities in the future, in the next 10 to 20 years, is AI-based technology, which we use in AI and semiconductor. So we thought, why not AI in education? So because India's launchpad to the global leadership, where most of you people are familiar, where all the, if you look at the Fortune 500 company CEOs, out of 500, more than 100 companies are headed by none other than Indians. And today, when you see the world's largest talent pool, that is from India. And the challenges what we face today in our education system is, so I still recollect my pharmacy days. So I studied my pharmacy way back, I completed 94. So still, when I interact with some of the students and some of the people, still the books which we read in 90s are still discontinued. The same books, nothing much changed, nothing, even the syllabus also. So now, the trend when we are talking about AI, when we are talking about semiconductor, when we talk about machine learning, all the new things, why not a pharmacy also can be included in these new technology things? So still we have a limited knowledge, AI faculty, we don't have a faculty who can able to, familiarize on AI and they can able to take classes. And low research output for the institutions. And also the biggest thing is the brain drain, which most of people look to go to US, and the good thing is in the last 1 year, the kind of the new policies, the strict policies of visa and other things. I think definitely it is a good thing for India, because graduates
what is the impact is graduates lack real work experience on AI skills, and only 600 professors with a deep AI expertise in the entire India. And we have 1,150 universities, 50,000 colleges, and more than millions of schools, but we have only that many members. And India ranked, now we are number 3rd in AI research. Definitely, this is going to take in a bigger way. So the research in academia is what is lacking in the education system. So that's how we need to create more entrepreneurs. That can be possible only through AI. Because if you look at the kind of the challenges and solutions, so we think about high cost and infrastructure, limited expertise, and workforce readiness. But we have a lot of solutions for that. We have a very innovative solutions which we come out, and that is the reason today these all investments are coming to India. Whatever companies I told, Microsoft or Google or Apple, because we have innovative solutions for us. So now the game-changing opportunity for us is the STEM education. So because India got an India AI mission, where 10,300 crore, the latest budget, last budget, they allocated only on AI education, whatever course it is. And all these investments, whether Microsoft, they are specifically, to give more skills on AI to youth. So that is a good thing, where we can get a lot of benefits out of that. So we must remind in education not just to learn AI, but to lead it. To lead it as a leader. That's how we can able to become. So this is one of the translating AI and ML research into industry scale innovation. It's not just about technology, it's about shaping the future of humanity. So the AI is going to make our work more simple, whatever work we are doing now. So educate, empower, elevate India through AI. So we are ready to help any kind of a solution you require in the area of artificial intelligence, any technology solutions you want to build any AI labs in your institutions. We at Vision Digital India are ready to support with Intel. Intel is ready to give some subsidies and options. So you can reach me, my email ID is there, my contact number, so anything if you require information on AI. So we are the company which working closely with more than the top universities in India, top government agencies, and some of the top technology companies. So with this, thank you very much for providing this wonderful opportunity to share something what exactly we are doing in AI. Thank you.
So if anybody has any questions, please ask.
The session is open for queries. If anyone, I request you to raise your hand.
Hello, sir. It was a really informative speech.
Thank you.
My question is that if I want to learn AI as a pharmacy student, where should I start from?
Start with the basics.
Yeah, I mean, it's like, yes, we know that we have to start from basics, but what platforms are available for me and what kind of topics I should concentrate on? Because normally for pharmacy students, they are weak in computer sciences and maths backgrounds. And AI is mostly related to coding and stuff. So it's hard for us to grasp it. So what kind of concepts should I learn first so I'm not terrified of learning AI and it becomes easy for me? And if possible, can you suggest me any such platforms?
Okay, sure. I think it is a good question now. I think you might have read some news where the government of India planning to introduce AI education from 3rd standard students from next year. Already NCRT is starting publishing the books, taking the syllabus from industry about AI. If 3rd standard student can able to understand AI, why not a pharmacy student? Because still I remember reading the Remington and so much bigger books. So AI is not only coding. So that is the other kind of a confusion, I can say, where everyone thinks AI means coding. No, it is not. AI is not coding. For example, when you are using a chat GPT, any coding you are doing? When you are using a chat GPT, so you are doing any coding or anything? You just ask prompt. You ask what information you need, you get it. So the similar way, for a pharmacy student, if you want to settle in the pharma industry, if you want to go to the research side, the best thing is learn some courses related to drug discovery, some courses related to the manufacturing process, how AI can be involved. Because today the drug discovery process is a very tedious process. It takes longer duration and millions and billions of money. So through AI, this process can be done faster. It doesn't require 20 years to develop a new molecule. So these kind of courses are available in so many international university platforms. For example, Stanford offer most of the programs at free of cost. Only when you want a certification from Stanford, then you have to pay maybe $30, $40 kind of a thing. Then you have courses in so many online platforms. Now, Indian government put a lot of information in Swayam. You can search Swayam platform, which is developed by IITs, where you have thousands of lectures on AI, including the practical experience. And the mission which I showed, you don't require coding at all. Even as school children can able to run this mission. So you have so many courses if you are really interested on AI. And you need not to be an engineer, computer scientist to do all this AI. That's how when we are talking of medical doctors. Today, all orthopedic surgery, 90% in India, is the hip surgery, knee surgery, is all happening with the robots. So what doctor knows, whether he knows the Python coding? No. They are able to use AI. So that is what. Thank you.
Thank you, sir.
Hello sir, good evening. By the way, it was really informative session. Sir, before times it was said like for freshers, you need a good experience to get a good package, and it depends upon how much you learn. But as AI has become more productive and various skill courses have been involved in the market, from last 2 to 1 year, it has been seen that freshers, irrespective of their experience, they are getting a good opportunities, and 50 to 60% of freshers are trying for their own startups using such ideas. So what's your take in this? As a fresher, you should go behind doing you something which you want to do for your startups as such in your initial stage, or do you think that experience still plays a vital role in professional life?
This is also, I think, very interesting question. Now I showed the product, which is developed by a 21-year-old student in his final year. Still is not completely received his convocation just a month back. And this product was launched 5 months back in Bengaluru by AACP chairman, one of Vice Chancellors. So today, in last 5 months, he installed these missions in 95 places. So each this device, the AI lab we call, will cost around 6 lakhs with a subsidy from India. So he made a turnover of closely around 6.5 crore in just 5 months back. Just 5 months old this product. And no prior experience, nothing. Just small idea which he is able to implement. And he taken a right guidance from mentors, taken a right advice whether this product will be scalable, whether he can able to make this impact, whether he can. Then the mentors given a proper guidance. They told you are not going to lose anything, just implement. So if that kind of ideas, if we can able to get, then you don't require a prior actually expertise or experience. But as I am in the industry for last 30 years, I started my career from one one of the top leading multinational company called Novartis. But always when you work with some good companies, you will learn a lot of good things. Because these companies are known giving their employees the best things. So if you work in these companies at least 1-2 years, you know how to run an organization. Because you are exposed to multiple verticals of business. So that will help you to scale up your company, whatever new idea you have, in a bigger way, where the failure rate will come down. Otherwise, what is happening is, we have plenty of ideas in India, but out of 100 ideas, I think not more than 4 to 5 ideas get into the market. So in my opinion, it is always, as a student, it is better to work for some good company for 1-2 years and start your idea and create an impact and create a wealth to the people. So that's how all these, new startups, like if you talk about Ola or Swiggy, Zomato, all these are with 2-3 years of experience, they're able to build these companies. So thank you.
Hello. Yes, sir. Already here, Government of India allocated 10,300 crores for subsidy for AI development.
Yes, it's not a subsidy, sir. It is the budget allocation to educate, to give AI skills to the students and colleges and
okay. So through the agency of AICT only to develop the AI programs.
Yes, AI labs, AI skill centers.
Sir, so you have shown the one lab, mobile lab now. What is the cost of that, sir?
This cost only 13 to 14 lakhs.
13 to 14.
Yes, because you don't require a brand new vehicle for this. Because you do painting for the entire vehicle, you do painting. So that's why you don't require a brand new vehicle. You get a vehicles. And most of our colleges and institutions, we have vehicles. One of the vehicles we can convert actually to this. And you require only the mission to run. That's
okay, with that mission, how much we can price it?
So, for example, if size of the vehicle is bigger, 30 to 40 students can assemble near the vehicle and they can get trained. So this doesn't require a huge number of minutes or hours. So you require just 1.5 hours to give around 8 to 10 experiments related to it. So this is the basic knowledge on AI.
Basic knowledge of it, AI, they can learn.
They can learn how
they can adapt in the pharmacy also.
Any course.
What are the knowledge they learn through AI training?
Yes.
They can adapt according to their will and vision.
Exactly.
Can you provide that?
Yeah, that vision costs just 6 lakhs.
Thank you for enlightening session, sir, and sharing your practical experience. Now I request Dr. Swati ma'am to hand over a moment to our renowned speaker. Thank you, ma'am. Thank you, sir. We are moving to the second session. I am very glad to welcome you, Dr. Srini Koli, sir. Sir is Vice President and Head and Sterile Product Development, Microlabs Limited, Bengaluru. Sir is having over 26 years of experience in product development, dosage designing, and modified dosage formulations.
Myself, Shreddy Kole, heading other injectables at Microlabs. Today's my topic is AI changing complex and drug product development. Before that, I just wanted to thank Scientific Service Committee for having me here. Particularly thank Dr. Mrs. Vandana Badlauri, who has cordially invited me to have this session here. Being in the product development industry for so long years, almost 27 years, that's the reason I chosen this topic of how this generic drug product development, particularly for the advanced markets, US, Europe and other geographies, how it is changing, and how the AI is helping in that. So basically I am going to cover only the industry related aspects, and I am viewing that angle of industry, what is happening actually in the research programs which are currently running in the Indian drug generic companies. So before move to this a topic of AI, I just wanted to convey, what is the economic impact of this AI in this generic world or the generic drug product development. If you see in the last few years, there is a tremendous transformation that has happened in terms of generic drug product development. So, and particularly the complex generic drug products. So if we see today, 90% of the prescriptions filled in the US, they are of generic. So which is giving a saving to the US government around 500 million, which is happening daily, which is an impact of almost $19 million. This is an old figure of 2022. This is kind of savings is been done by the US government and majorly contributed by the Indian generic companies who have exported this kind of products to the US market. So if we see kind of opportunity which is there going forward, which is almost 25 billion, more than that, and if we see the first 10 products in the market which are coming, or first the 10 innovator products which are having, almost more than 25 billion as a market opportunity. So Indian generic drug companies, are having this kind of targets to get those kind of market in the US. So the pharmaceutical industry is shifting from simple oral dosage forms to complex injectables, and particularly the peptide based and biologic derived products. So if you see recently also, there is a lot of hype regarding the GLP-1 products like semaglutide, tirzepatide. I am going to cover this in my subsequent slides. So if you go back to the era of 2000 and 2005, there are a lot of oral drug products. So right now the focus is shifting more towards the complex injectable drug products. So as I mentioned, GLP-1 agonists represent $1 billion market opportunity for all the Indian generic pharma companies, and in which the AI is enabling the Indian companies to move it faster, get the drug product development faster, and get it approved and get it commercialized in the US. So I'll just briefly cover about the history, like how the industry has been moving from 2 decades. If you see the last 2 decades, moved from the simple oral tablet capsules, which was around 70-80% of the market way back in 2005, which has now been declining the share and becoming kind of a commoditized with the minimal profit margin. So my entire talk is with respect to the generic pharmaceutical industry and how they are viewing that. Now, if we go back again to the 2005 era, it's a solution products, injectables, which is constituting around 20%, and now there is a very modest growth where non-complex parenteral solutions remain important but stable as a market, if you can see. So where is the main crux, where is the shift is happening in the complex generics, where less than 10% of the market, primarily limited to topical products way back in 2005. You might be knowing this. There were no product-specific guidances for those kind of products. All were required to do the kind of clinical studies. Some differentiated products also required to do the clinical studies. Now it has been moved towards complex injectables, which constitutes around 20% of the market share, and particularly the explosive growth has been given by GLP-1 drugs like semaglutide and tirzepatide, which are mainly for the diabetes and weight loss management. So this dramatic shift reflects the pharmaceutical industry's evolution towards more sophisticated drug delivery systems and the economic imperative to develop affordable, alternative, expensive branded drug products. So what are those complex generics? When we are talking about complex generic drug products, what are those? So I have bucketed them into four. If you see peptide-based medications, like I mentioned about GLP-1 drug products, particularly about the diabetes and weight loss management, semaglutide, tirzepatide, liraglutide. So these are the kind of molecules developed by Novo Nordisk and Eli Lilly. They are making the wave in entire industry or entire globally, I would say. So these are the complex drug products which have been characterized by their variable molecule structures, post-transitional modifications, immunogenicity risk, multiple molecular species in single products, and potential aggregation during storage and delivery. So these are the certain characteristics which then make them complex. So if somebody wants to develop these kind of products, they need to overcome these kind of challenges, particularly the potential aggregation and the immunogenicity challenges in development of those particular products. The next would be the long acting injectables. We can talk about liposomes or microspheres, which formulations with dosing intervals of almost 3 to 6 months required extended PK studies, and certain these are the complex depot formulations. So again, in terms of the complexity, I mentioned variability from injection technique and challenging the release kinetic predictions. So these kind of attributes of these dosage forms make them really complex, and it's very challenging to develop those kind of products, which is again similar as that of the innovator. Third would be inhalation products, again DPIs, MDIs, which is again a kind of a complex products, which make them complex in terms of their formulations as well as devices. So each if you see the DPI, MDIs devices, each has been patented. There are hundreds of patents of each of the devices, which has been lasting more than 20 years. So which really makes the Indian generic companies very difficult to replicate those kind of products, which difficult to generalize those products. And fourth bucket would be drug device combinations. So again in that bucket, there are multiple products. It can be small molecules, it can be peptides, it can be biologics. So drug device combinations, which mainly constitute the device component, which is important, like pen, where pen has a multiple doses design. So suppose if we talk about the semaglutide, it's a weekly once dosing, and there are 4 doses in a one single pen, which is given by the given to the patient. So that is a pen. Auto injectors, like suppose single dose has been to be given to the patient. So that's an auto injector where the spring brace design is there. So which makes them very difficult to develop and test also for the Indian generic companies. So coming back to our topic of AI, how this AI is helping the generic Indian drug companies, and what kind of tools and techniques being used by these industries to make this product development faster and really able to launch those kind of products early in the US market. Before that, just to summarize, I think this morning many speakers have spoken about what is AI, what is ML, what is deep learning, what is generative AI. So I will not go in explaining about the definition of those. You might have known everything about it, but I will try to cover about the practical aspects of it. So or how the AI is going to help us in the drug product development. So these are some of the areas like molecular predictions. So some speaker was talking about the new chemical entities. So how these new chemical entities getting developed, and in that which kind of AI tools, techniques being used, which will help us to reduce the process of getting the new chemical entities. Formulation optimizations. When we are talking about the complex generics, there are a lot of challenges in terms of stabilization of those products, particularly the peptides ones, where a lot of challenges is in terms of forming aggregations, impurity profile. All those challenges can be overcome by using certain AI tools, which I will cover in my subsequent slides. Bioequivalence analysis. So normal generic industry, every product required a bioequivalence test, and there are multiple iterations to be required to make this product bioequivalent to the in comparison with the innovator or the R&D, we can say. So to reduce those iterations, to reduce those timelines, to reduce those tests to be carried out on healthy human volunteers, there are certain AI tools which can help them to reduce those iterations and make them predictions about the formulations which has been made by the generic companies. So without testing on the healthy human volunteers, it can be gone ahead for the subsequent batches. Manufacturing excellence. So multiple people have covered again in terms of process optimization and quality control, what kind of tools, techniques are there. PBPK modeling, this is advanced pharmacokinetic simulations, which will help us to develop certain differentiated products where innovator is there in the market, but if you want to make a differentiated product, so you're changing your formulation compositions in such a way that there is a need for doing the clinical studies. So without doing those clinical studies, how this PBPK modeling AI tools help them to mitigate those kind of risks in terms of carrying out those clinical tests that we will cover. So AI tools revolutionizing the drug development, and which are the certain industry standard platforms enabling this complex generic development. One would be Schrödinger suite. So this is an industry gold standard for computational chemistry, which this different modules suite like Desmond, Clyde and Jaguar handle different kind of applications which will suitable for making the new drug discovery. So there are what essentially they will do, they will scan multiple molecules and help to reduce the development time. PBPK modeling tools, as I mentioned, certain differentiated products requires change in the formulations, which requires again clinical trials and all. So tools like PKSim, SimCap simulator, GastroPlus, this will help to predict the bioavailability of the complex formulations, and these we can adopt and make our product similar as that of the innovator. So these platforms perform population-based PK simulations incorporating patient variability, and with the support of these approaches, we can reduce the cost almost more than 20% of the traditional approaches which has been normally followed in the generic drug industry. Also, the development timeline also can be reduced considerably. GastroPlus, it's a very innovative, very novel tool in terms of modeling and simulation package, which will help us to predict the bioavailability of the drug products. So in terms of bioequivalence, this is a very great tool and which is very commonly used by the generic drug industry. Another machine learning platform which is by DeepMind is AlphaFold derivatives, and this custom machine learning model predicts structural protein and peptide 3D structures and stability of synthetic peptides. So this is very particular and very useful in peptide drug product development. Each of these molecules, like suppose semaglutide, it's around 31 amino acids and been synthesized either by synthetic way or by recombinant DNA way. So these kind of tools and techniques will help us to shorten the development time in terms of predicting these 3D structures and how the stability of the synthetic peptides can be maintained. Random forest is another type of machine learning model that uses an ensemble of decision trees to make its predictions. So this is another tool available for the regression task, predicting drug activity, toxicity, and patient response. The next one is expose algorithms perform drug response and IVIVC predictions. So particularly these IVIVC predictions, as you know, drug classifications, I think it's been taught in first year maybe, like solubility versus permeability. So particularly the class 4 drug products where there is a very less solubility, very less permeability. These kind of drug products are very difficult to develop and very difficult to make the bioequivalent product as that of the innovator. So in those kind of drug product developments, this is very useful in predicting the or making the IVC correlations, where you can use your in vitro techniques and predict its performance in vivo without doing any kind of studies on human volunteers. So, what are the AI solutions for complex generic drug development, and how the FDA, and I'm saying FDA, it's US FDA particularly, and what kind of platforms they have, and how this regulatory pathway for complex generic drug products, particularly the peptide and biologics, is evolving. So FDA has deployed 3 critical AI-driven regulatory tools that streamline approval pathways while maintaining rigorous safety and efficacy standards. And the first one is clinically informed meaningful differences, CIMD detections. So what it means, it analyzes Q1, Q2, Q3 differences between those drug products with different orthogonal techniques. So that Q1, Q2, Q3 differences between your test and RLDs has been reviewed by the AI tools and FDA to find out clinically informed meaningful differences. So it means if it has any differences which are irrelevant, or which are very marginal, then it doesn't require additional clinical studies. It doesn't require additional orthogonal techniques to prove that your product is bioequivalent. For example, in ophthalmic products, AI fingerprints excipients composition differences and applies PBPK models to predict bioavailability impact. And if predicted differences is less than 10%, biowaiver is granted. So that is how the US FDA is moving towards screening the applications, reviewing those applications, adopting those kind of tools and techniques while reviewing those drug products. Second would be the safe harbor justifications for formulation flexibility. So its AI-driven risk assessment supports 21 CFR with justifications enabling manufacturers to take minor formulation adjustments without repeating costly BE studies. So as I mentioned earlier, if you make a kind of some differentiated products and you made the changes in your either in the surfactant or buffer or the pH or any of the tonicity agents, that doesn't require to completely repeat those kind of studies again. So how you can do that? With this help of AI models, predicting less than 10% PK impact and allowing excipient changes, providing sourcing and manufacturing flexibility. That is how you are making your product similar as that of the innovator drug product. The recent one is peptide oligonucleotide submission de-risking. So as I mentioned, it's a very peptide and oligonucleotide, it's a very complex kind of drug products, right from API manufacturing to drug product manufacturing as well. So if you start assessing your risk in terms of immunogenicity, aggregations, any changes in the structure of the molecule, if you start assessing those with the AI tools, instead of doing the